पूरा सेशन इधर ही रहता है।
नहीं, हाँ 11 साढ़े 11 बजे तक यहाँ फिर ऊपर यही सब सेट करिएगा। कैसे भेजो? मेल में भेजो।
हाँ ये इसमें आ जाएगा।
मिल जाएगा। भाई एक टेबल मेरे को भी लगेगा।
सबसे बड़ी भी लेके जाना है। क्या चाहिए? नीचे स्पाइक गार्ड है, आपको लगेगा था ऊपर जगह बचा नहीं, स्पाइक गार्ड लगा रखा है ऊपर तो नीचे तक तो आई है। इधर में तो चार्जिंग का वायर आ जाएगा।
चार्जिंग का वायर आएगा।
I'll show you what is what.
Excuse me. Getting can you share this link to WhatsApp? Scientist group.
I'll change the name for it.
You do that one here. No issue. Sir, Mukunda sir. Recording is done, sir. Recording is done. यूज करेंगे उसको नहीं करेंगे इसलिए तो मैं पूछ लिया बाद में
नहीं बाकी लोग पूछ रहे हैं कैसे एक सोफा यहाँ पे लगा देते हो आप कैसे यहाँ से एक सोफा लगा दो सोफा लगा देते
ये इसमें लगाता हूँ माउस को। हाँ हो गया ना सेटअप अपना पूरा। हेल्प करो देखना ये लो।
कहाँ रखेंगे इसको? इसको भाग ले रहे हैं।
नहीं
अच्छा वो अपना वो स्पीकर का उसके आगे
लगा
आया लगा दिया। हाँ सर ब्लूटूथ में चाहिए क्या?
Fisheries Biotechnology. The quest for cultivated meat as an alternative supplement to conventional meat, viable alternative smart protein for the generations to come. Taking this forward, we are honored to have with us as our esteemed chief guest, Dr. Anupam, Deputy Director General and CAR Constituent Event. I humbly request our Director and Vice Chancellor, Dr. Ravi Shankar sir, to do the honors of welcoming our chief guest. We also have with us today our distinguished guest of honor, Dr. W.S. Lakra, former head of the CIFA family, former director and vice chancellor, ICAR CIFA Mumbai, and the secretary of National Academy of Agricultural Sciences, New Delhi, India. I now request Dr. Mukunda Goswami, Head Fish Genetics and Biotechnology Division, ICAR CIFA Mumbai, and the convener of this international conference to please welcome our Honorable Director and Vice Chancellor, Dr. C. N. Ravishankar sir, and our Joint Director, Dr. N. Science. I request Ms. Kriti now to welcome Dr. Mukunda Goswami. I request Dr. Mukunda Goswami to please welcome our Joint Director, Dr. N. Science. And I now request Ms. Kriti to please welcome Mukunda Goswami, Dr. Mukunda Goswami, the convener of this international conference on fisheries biotechnology. Keeping up with the Indian tradition, I request the dignitaries on the dais to please proceed for lighting of the sacred lamp, commemorating the auspicious beginning of this 2-day-long scientific communique. Thank you, sirs. I hereby take the privilege of bowing down to the Lord Felicia with this invocation melody. Shankara Subhadam Bhavani Nandana Shankara Subhadam Bhavani Nandana Gai Ganapati Jalavandana सिद्धि सदा राज वदना विनायक सिद्धि सदा राज वदना विनायक सिद्धि सदा राज वदना विनायक कृपा सिंधु सुंदर भवनायक कृपा सिंधु सुंदर भवनायक गाई गणपति जगवंदन गाई गणपति जगवंदन मोदक प्रिया मोदक मंगल मोदक प्रिया मोदक मंगलदाता मोदक प्रिया मंगलदाता भाग्य विधाता वृद्धि भाग्य विधाता गाई गणपति जगवंदन गाई गणपति जगवंदन शंकर सुवन भवानी नंदन गाई गणपति जगवंदन गाई गणपति जगवंदन. It is my request now to our Honorable Director and Vice Chancellor, Dr. C. N. Ravishankar sir, to kindly present the formal welcome address.
Good morning to everyone. Most respected, the chief guest for today's conference, my distinguished scientist, my dear friend, the Deputy Director General of Animal Science, ICR New Delhi, Dr. Raghavendra Bhattaji, and our own former Vice Chancellor, the Director of CIFE, former Director of NBFGR, the Secretary of Finance, and our own family, Dr. Lakra Sahab, Dr. Sahu, Dr. Mukunda, the convener of this conference. I see the galaxy of very senior people. I see Dr. George John, the former senior advisor to DBT and the Vice Chancellor, Dr. Ponaya, the former Director of NBFGR, Siva, former from the World Fish. I see Dr. Linave Sahab, the former senior advisor to DBT, my friend Dr. Vijayan, Dr. Girish Patil, I see Dr. Vincent George, and our own teacher, Dr. Diwan Sahab. And I see many, many other invited distinguished speakers who have accepted our invitation. All the delegates of this conference, my dear students and all the faculty, a good morning to everyone. First of all, a warm welcome to all of you to this institute, the Central Institute of Fisheries Education, and also to City of Mumbai and for this conference. I think we just completed, the Asian Fisheries and Aquaculture Forum in Delhi recently. So all of us met there. So in fact, this conference was supposed to be held just before some time. Then we thought, let the Asian Fisheries be over. Then we thought we should have this conference. So that's what we have. We are here today for this 2 days conference. And I'm sure I will not go into the details about this conference. Mukunda, the convener, will be talking about what are the themes, what are the technical sessions which we will be having for the coming 2 days. But let me take the pleasure to invite and welcome all the distinguished people here formally. And it's a great pleasure to have all of you in CAFE. Being an education fisheries university, it's always our endeavor to bring in all the senior people to this institution so that we can have more deliberations, more meaningful dialogues, and all the senior people share their experience and expertise with our students and our faculty so that we are updated all the time in all the relevant subjects, even though we are talking about the biotechnology today. Biotechnology is not specific to any specific area, it encompasses so many areas. And just briefly about my institution for the people who have come from outside. This is a university which was started as an institute for giving training to the department officials by UN nearly 62, 63 years back. So it was basically a training institute, then it got into the Indian Council of Agriculture Research, then it became the deemed university and the UGC. So we are the Fisheries University, the first Fisheries University under the Ministry of Agriculture and Farmers Welfare, where we are offering masters and PhD in 11 disciplines. So one thing is we are not offering the undergraduate program so far, but probably when we are asked to comply with the new education policy of Government of India, so maybe in the future we will be looking at starting the UG program, which is mandatory for any university. From upgrading ourselves from the deemed university status to the full-fledged university, we may have to do many things, some of the things which we have already started. For example, in addition to the higher education which we are offering to the masters and PhD, we are already offering the diploma and certificate courses, especially some of our centers. In addition to the headquarters where we are here, we have 5 centers, one in Haryana, in Rohtak, one in Motipur in Bihar, one in Calcutta in West Bengal, then in Kakinada and Balaputrapuram in Andhra Pradesh, and we have one in Pawarkheda in Madhya Pradesh. So where we are offering the diploma and certificate courses, the need-based programs, skill development programs, a lot of human resource development programs, and focusing now recently on entrepreneurship development programs, in line with the Government of India initiatives where the government is also supporting the startups, the entrepreneurs. So that's what this institute also is doing. That in addition to giving the quality human resource to the sector, we are also giving other skill development programs for the sector. And I'm sure all of you know, when fisheries sector, which is growing at unparalleled to any other sector, I think we should all be proud of that we are all part of the fisheries sector. Compared to any other sector, we are growing at above 8%. So we take pride in that, but at the same time, a lot of expectations from institutions like CAF, where we have given the quality human resource to the sector. Whether we are talking about the aquaculture sector, we are talking about the nutrition, we are talking about the health, environmental management, the post-harvest technology, at the end of the day, connecting people through our social science initiatives. So whatever we take, any domain of fisheries you take, we are the people who are giving the quality human resource, and the graduates who are coming out of the institution are joining as a scientist or going to the departments, they go to the universities, then they also get into the private, starting their own enterprise, they go abroad. So they're all making us proud. So at the same time, when the sector is growing, naturally the demand for the skilled human resource is also increasing. So that becomes our responsibility to give the quality human resource. That's what we are here. For that, we believe that having conferences like this, bringing people together under one roof, creating a platform for creating synergy between the industry, academia, and institutions. It is not just specific to the state departments, we are also talking about the developmental agencies. So we take always, a lot of initiatives to bring in all these people, give a platform, connect the bridges, collaboration, partnerships, that's what we are all looking at. So in that direction, this conference also is a very milestone for CAFE. And I would like to compliment Dr. Mukunda and the whole team of the Genetics and Breeding Division, Biotechnology Division, for taking this initiative. And I would like to compliment the whole institute. There is not, when we talk about any initiative in the institute, it is a teamwork. And CAFE always believes in CAFE Parivar. So that's what we always believe. Whatever we do, we always do as a teamwork. So that's what we are here to do that. And also just briefly telling about some of the initiatives which we have taken recently. Because when we talk about the human resource, what is important is our connect with the state departments, with the universities, and other departments. So what we did religiously, annually, we bring in all the deans of the fisheries colleges here. We talk to them how to improve the curricula, what courses we should be doing. So we have always in connection with the deans and the vice chancellors of especially the fisheries universities. In addition to that, we bring in all the directors of the state department, the state fisheries departments, where we can do the hand-holding with them, where we can help them in, because every state department has their own limitations. whereas we have the quality skilled experience here. When I say CIFE, I talk about all the fisheries division. We talk about 8 institutes. Again, it's a theme we always talk about. We don't talk CIFE as a standalone institution. So when we say, we talk about all the 8 fisheries institutions. So we are always connected with them. Our students go there, their faculty are our, the adjunct faculty, and we have recognized 100 scientists across the 8 institutes as our major guides. So that is how we are always having connection. That's how we can build, we can stay relevant in the sector when there is so much of expectation and demand from the sector like this. So that's what we see. We are always taking initiatives. So in addition to that, we want to promote student entrepreneurship. Every time, every year, even though we have our own mandate, in addition to that, we take every year a theme, like commercialization of our technologies, improving our partnerships, improving our collaborations, and also promoting student entrepreneurship. That has been our major focus. So we have been doing that, wherein we take our students as incubators. We also run an agribusiness incubation in this center. So we take the students and we promote them. And when we do all this, we bring the students of all other fisheries colleges. And last year, we started another very good initiative of the recruitment drive, like similar to any IT or other BT people do. So we brought in all the fisheries college students here. We brought nearly 60 companies here. So we created a platform for them to pick up the good talent from all the fisheries colleges. So that was the first initiative which we hope to continue in the coming years. In addition to that, fish is very healthy and nutritious. I'm sure all of you agree. So that's why the fish sector is growing so fast. So promoting fish consumption is also our responsibility. So for that, we are making, because in an institute like CAFE, we have students across India. We have students from Kashmir to Kanyakumari, from Gujarat to Northeast. So the fish delicacies are different. People likings are different. So how we can improve the fish consumption? Only by providing what they like. We can't force somebody to like the Kerala cuisine who is in Kashmir. So what we did, we create a platform for all the students to prepare the delicacies across the pan India, and that day we keep it open for the public. So last 3 years we have a tremendous response on this fish. We call it as fish swap. So that past 3 years that has been a big hit in Mumbai. So a lot of people come here, enjoy the delicacies of across the country. So that is one way of promoting fish consumption. When fish consumption improves, the fish production will be improving. So at the end of the day, we are going to help the fishermen or fish farmers, all these people. The whole sector, that's all the stakeholders will get benefited. So CIF is not just restricting ourselves to the higher education. We are always connected with everybody throughout the sector. So that direction, we are fortunate to get the expertise and experience of the senior people. So I'm happy that accepting our invitation, very senior people are here. So once again, a warm welcome to all of you, and we want you to be connected with us, continue to be mentoring our students and our faculty, so that we can be relevant and you can be more meaningful in what we are doing. Because the future days are not going to be there we can work in silos. We need to be working in partnerships. Every time we are talking about multidisciplinary, multi-institutional things, whatever we are talking, any projects we take. So that way we are happy that we are getting people from Indian Institute of Science, CCMB, so many institutions have accepted to be part of our conference here. So we are looking forward for a very vibrant, very informative, very enriching 2 days of conference. And personally, I would like to thank Dr. Bhatta, because the parliament session is going on. None of the ICR officials are permitted to leave the headquarters. Dr. Jana could not come. Once again, on this occasion, I would like to place on record all the support by DDG Fisheries and the Secretary there and ICR and DDG Education. This institution, even though we are under the subject matter division of fisheries, we do have a lot of connection with the education division. Because where we get the funding, we get a lot of support. And one thing I would like to tell you, we are going to expand our activity, we are going to improve our old campus, we are going to have vertical expansion of this building also, and we are going to strengthen a lot of activities for the students. So all that was possible only with the support of the Indian Council of Agricultural Research, the secretary there, and the DDG Education, DDG Fisheries. On this occasion, I would like to place on our record, thanking them for all the support. And on this occasion, I would like to again warmly welcome Dr. Bhatta, my friend, for this conference, for taking time to come here and to be with us, because we don't feel again we are part of other than the animal sciences. The fisheries and animal sciences are always together. So we can do a lot of joint programs together, because a lot of disciplines are overlapping. And you can see a lot of animal science scientists working here for so many years in this institute. So we are looking forward for all the support, Dr. Bhattaji, and a warm welcome to you. And of course, I don't have to tell about Dr. Nakra Sahab. So he has been part of this institution, he has been supporting, he has been mentoring this institution. A warm welcome to you, Nakra Sahab, for this conference. And I welcome everybody, all the delegates, all the invited speakers, all the chairs, co-chairs, all the delegates from the outside, all the students from other institutions. So it has been a great response for us. When we started off this one, there was an overwhelming response. So I'd like to thank all of you for coming over here. And I wish you a pleasant stay here for 2 days. After this conference, in between, please take time to go and see our other divisions, go around the facilities, what we have. And Bombay is a very happening place, so you can go and explore the city of Bombay also. Once again, a warm welcome and complimenting Dr. Mukunda and the whole team, and not everybody, all my colleagues in CAFE, for putting up a grand show like this. And we look forward for a very 2 days enriching experience. Once again, warm welcome to everybody. Thank you. Jai Hind.
Thank you very much, sir, for extending a warm welcome to our most respected galaxy of guests, highlighting this event with its visionary theme, which we believe will push the limits of science towards higher territories. Today, we have gathered here especially to celebrate the quintessence of this international event, to discuss global advancements in biotechnology and its crucial applications for vitalizing the Blue Revolution mission in aquaculture and fisheries. Therefore, I call upon Dr. Mukunda Goswami, Head Fish Genetics and Biotechnology Division and convener of this international conference, to kindly present an outline of this 2-day-long scientific communique.
Thank you. Good morning to all present here in this August hall. Chief guest of the inaugural program of International Conference on Fisheries Biotechnology, respected Dr. Raghavendra Bhatta, Deputy Director General Animal Science, ICR New Delhi. The guest of honor, respected Dr. W.S. Nakra sir, former Director and VC of CIF Mumbai, and currently Secretary, National Academy of Agricultural Science, New Delhi. The respected Director, Dr. C.N. Ravi Shankar, Joint Director, Dr. N.P. Sahu, all the esteemed guests occupying this August hall, Dr. George John, former Senior Deputy Advisor and the Vice Chancellor, Dr. A.Z. Punia, sir, former Director at SIBA and NBFGR Lucknow, and Dr. A. Ninave, former Senior Advisor DBT and Vice Chancellor, and Dr. A.D. Diwan, former ADZ, Dr. Greenson George, Director CMFRI, Dr. Girish Patil, Director NRC, and Dr. D.K. Das from Director CP will join soon. And I can see my teacher and many senior colleagues like Dr. Sanjay Das and many other distinguished scientists and students from all over the country. And then I would like to greetings Dr. K. Khandaras, the Director CBFT, and Professor B. Gunul Gopal from ISC Bangalore, and then esteemed keynote speakers, chairs, co-chairs, invited speakers, lead speakers, sponsors, and the distinguished participants, guests present here. The organizing secretaries, Dr. S. Agidar and Dr. N.S. Nagpuri, the faculty members of CIFE and my divisional team, the representative of various organizations and industries, students, farmers, media persons, ladies and gentlemen. It's my great pleasure and honor to welcome all of you to this international conference on Fisheries Biotechnology being organized by ICR Central Institute of Fisheries Education Mumbai in association of Indian Fisheries Association Mumbai. Sir, at the outset, we are extremely delighted and deeply honored to have the Deputy Director General Animal Science, Dr. Raghavendra Bhatta sir, as a chief guest of this tourist function. Sir, we express our highest gratitude on behalf of ICRCF in Mumbai and all of us present here. And thank you so much, sir, for gracing this occasion as a chief guest in spite of your acting civil and political mention about the party administration. We extend our welcome and deep gratitude to honorable guest of honor and head of CIP family always, Dr. W.S. Lakra sir, who has been on the forefront of the fisheries sector, particularly fish biotechnology, and you'll be happy to know that sir has been figured among top 2% of global scientists as per Stanford University, USA. So I don't have words to talk about sir, as well as you know, his contribution is like awesome, and I am not no way to talk about him here. And then he has been advancing all the premier institute like NBHR as a director and Central Institute of Fisheries Education as a director, and two like world-class institutes, advancing these institutes. And today, the beautiful campus we are here, it is his great contribution only during his tenure when he was in CIP as a vice chancellor. Sir, now as Sir again, sir is very active. Recently, he organized Indian Agricultural Science Congress in Zircon University with grand success, where maybe 3,000 participants were there, with maybe more than 20 international organizations. And then in addition to that, he organized one more Indian Science Congress in Kochi. So, sir, there are so many things to talk about you, but it's today I am, any addition I want to say today is truly is because of Sir's guidance and blessing I am standing in front of you. If I am doing any good work, it's just because of him. Thank you so much, sir, for gracing this occasion as a guest of honor. We are organizing this conference with all the overall leadership of our director, sir, Dr. C.M. Ravi Shankar, sir, and the President, Indian Fisheries Association. And then the leadership, really, he is giving us, motivating us. And thank you so much, sir, for your support and leadership. We are here today because of hard work done by entire CIT faculty members. It's a very happy to say each one of the faculty members, staff members, students are contributing to this conference, particularly my exhibit team, day night, and huge support by all of you here, giving your support, consent to be here. This is a huge gathering, and this August hall is full with several stalwarts, guest experts from India and abroad. Will be getting connected with some international speakers and many offline speakers and participants. So many international speakers expressed their interest to be here, but I could not make it because of some formalities or ministry level clearance. But next time we'll be trying to make it happen in physical mode also. So I will be failing my duty if I don't acknowledge the Lakra sir's contribution in organizing the first conference in Fisheries Biotechnology in 2000, and then second one in 2012, and then third one we are here under the leadership of Dr. C.M. Ravi Shankar sir, and this is the after 12 years, the first and second 12, and second and third is 12 years. Sir, as we know, I want to talk a little bit about the subject behind this and about the conference. As you all know, by 2050, we will be 10 billion people. For a 10 billion people, managing food is a huge challenge. And then we have another SDG by 2030. And then we know that we are going to be Dixit Bharat by 2047. If all this to happen, again, as per UN estimation, we need to produce 70% extra food. So where from this 70% extra food will come? So that's what the CIB has also initiated, prepared for this Dixit Bharat by initiating a cultivated seafood program at international level, and CIB is leading the program. And many other international institutes are collaborating with CIFE. That's what and that project was funded by Good Food Institute USA. Then we have gathered here to discuss strategies and to form a blueprint for the fisheries sector and the role of fisheries biotechnology. And then under this, we have around 10 technical sessions, and there will be some sessions will be going on parallelly. And to cover the theme of today, this theme is biotechnology for future seafood, the smart protein, alternative protein, and the blue revolution. So you all be fortunate to hear from the eminent scientists as keynote speakers, invited speakers from different institutes of the eminent institutes like Indian Institute of Science, Bangalore, 3 IITs, IIT Bombay, IIT Madras, and IIT Guwahati, CSR, CCMB, DVT Institute of Life Science, ICMR, NIRRCH located in Mumbai, DSC Agarkar Research Institute, TIFR, and BHU, DVT, and including our distinguished scientists from ICR institutes, and at the international level, there will be many speakers. There are keynote speakers. First one will be from Argonne University, USA, Canada, Singapore, Netherlands, Australia, Norway, and many more will join online mode. And then altogether we are with this will be helping by will be getting help from GFI, and altogether we are at 300 registrations, and many more will join in this August month, and then maybe many more will be there in online mode also. We'll fortunate to hear from all these 3 speakers, keynote speakers, and 30 invited speakers from national and international organizations. There are academic sponsors, TSC, DVT, EMPEDA, and NFDB, and there are some industry sponsors, numbers of more than 10. High media, particularly in Scientific, and there are knowledge partners, CFI, Woodford Institute, IIT Bombay, and CIT Alumni Association. Sir, I would request all of you to kindly make use of this conference and have a fruitful discussion, deliberation, and we look forward to your active participation, guidance, inputs, and I am sure you will enjoy this conference and get new insights, knowledge, and also your comfortable stay in this beautiful city, and maybe we can be a part of Viksit Bharat by 2047. Thank you very much. And then I just with a Sanskrit slope, Sarve Bhavantu Sukhina Sarve Santu Niyame. Thank you very much.
Thank you very much, Dr. Goswami, for presenting the genesis, a brief introduction and outline of this conference. Today, we are ecstatic to have among us Dr. W.S. Lakra, our former head of the CIFE family, former director and vice chancellor ICAR CIFE Mumbai, and secretary National Academy of Agricultural Sciences, New Delhi, India, as our distinguished guest of honor. An eminent scientist and professor of international repute, he has held prominent positions throughout his career. As director of the ICAR National Bureau of Fish Genetic Resources, Lucknow, followed by director and vice chancellor of the ICAR Central Institute of Fisheries Education, Mumbai, the NABARD chair at the ICAR CMFRI.
Thank you. Chief guest of the function, on the stage, friend, and the DDG in Science, Dr. Manmeve Gupta, Director, Vice Chancellor of this great institute, Dr. Shugarji, Dr. M.P. Sahu, Joint Director, and Dr. Goswami, convener of this international conference. And all these teams, now delegates, guests, participants, who have been named by many earlier speakers. Very good morning. For me, this is one of the happiest occasions that I am with my family here. And very happy to be a part of this international conference. And I'm meeting some of my colleagues from the system after a very, very long time. And I can see here our seniors. I always tell them my seniors, Dr. George John, Dr. Kinaya,
डॉक्टर दीवान, वो मुझे एक कहावत याद आती है, ये अलग बात है कि वो अलग खड़े हैं, ये अलग बात है कि वो अलग खड़े हैं, लेकिन वो बड़े हैं, वो बड़े हैं। ये हमारे बड़े और प्रोफेसर धनराज, former director of CDFP, now scientist from Gopal from IC Bangalore, Dr. Vincent of Patel, Dr. Vijayan, our former friend in our Director Siba, and all the HODs I can see here, Dr. Ketkar, Dr. Nenave, a very popular name in the biotechnology, and everyone present here, students, friends. I think let me first compliment the institute, Dr. Ravi Shankar, the entire team for the achievement of this institute. We've already passed, we will be completing shortly 75 years of it. So the institute from a training center to this center of excellence in higher education itself, I think this is a marvelous achievement. And as we all know, the Rome was not built in a day. So this has taken this much time for the students. I remember I was going through this brief profile of the department. And this reminds me when we started the biotech journey at this institute, somewhere in 1987. The then director called all the young scientists. The youngsters should know that. He called all the young scientists and he asked them to define biotechnology. It was somewhere in 1987. And none of us were able to define biotechnology. Many of the students are not able to define nowadays also, when we ask them in the interview board. But then at that time, we were completely ignorant. But the then director of the institute, he was with FAO and he was running a unit program. So then he said, he identified one scientist that I'm making a laboratory or a section of fish biotech and high-tech systems lab or a section. With no high-tech, no biotech, but a room was given. And I was fortunate to be selected for that, identified for that. And I had no expertise in biotech. So that was the journey. And then we started running around to know who are the people who are working in this area. That's how I said Dr. George John and Dr. Polier. They were already there in this. Dr. John's earliest publication on gynegenesis and Dr. Polier's work on molecular genetics, they have already started in that country. So we all started running around to learn about genetics. And we went to all places, including CCMB and this one, our MKU, Madurai, Kamra, University, Professor Pandyan. And I think Dr. Das is here, our great friend, Dr. Das. Those days, we all attended programs of chromosome manipulation at the MKU, 3 weeks program, including Dr. George, Dr. Pune. Everybody went there. All those who were interested in fisheries biotech. So that was the big. The rest is all history. And we had only, initially for several years, we were only 3-4 scientists. I know myself, Dr. Jagadar is here, he's organizing secretary, and doing the Vishnu, Dr. Parna. So 3-4 scientists for several years. Even we started this all Dr. Shankar mentioned about biotech programs and research. And now to see this profile, you have worked as a scientist now, in partnership with leading organizations like CCMB or IITs and ISCs. It's commendable. Dr. Goswami, we compliment you for your achievements in the inauguration. Our association with CCM, at that point of time when we were developing our skills, maybe Dr. Thangraj is here and Dr. Lajji Singh, Dr. Lajji Singh, they were the leaders for DNA printing and DNA technologies. So we all were so excited to know what is this all about. And we developed our contact with Lajji Singh. And when we said Dr. Lajji Singh, Dr. Thangraj was the next person with Dr. Lajji Singh. So that was the team working there. And we had a lot of interactions with Lajji Singh came here several times. And we got training there at CCM. Thank you, Dr. Thangraj, for making it today. And so that's and of course, Dr. Majumdar was there earlier. And let me also share with the students, this excitement in fisheries biotechnology in those days, but not with the fisheries institutes. CCMB had a program on the transgenic fish and things like that. And I had a program on transgenics. The first training course on transgenic fish production at NI in Delhi. But then they were definitely shocked, Finally, the fisheries institute took over because it's our mandate. The mandate of the ICR Fisheries Institute. And the actual research boost for fisheries biotechnology was given by DBT. The department was created in 1986 or so. And for the next 25 years, I think, it is DBT funded programs, whether it is the animal sector or the crop sector or fisheries. What you see today, whether the human resource or the research program projects or the centers of excellence, is all funded by DBT. We remember Dr. John and Dr. Inauguration. It was so positive. And they were, in fact, taking this lead across the country. It's very unfortunate now that is not the case now. That kind of support we are not getting from DBT. But then we have become strong now. The ICR Institute, in every ICR Institute now, you have the expertise, you have best of the facilities, you have developed partnership, national international partnership. So what all you need? You need only a kind of a more focused, approach, targeted approach, innovative approach to biotechnology. I think that's what this conference should be. And let me also tell that the first task force in DBT on fisheries and aquaculture and biotechnology was chaired by Dr. S.M. In the morning, I was having a walk and I saw Dr. S.M. aquaculture complex. I was compliment Dr. Joshi. You have very rightly named this facility. I remember when he was the task force chairman, he used to visit institutes and he was asking us, give us projects on biotechnology. What we were not knowing what is biotechnology. And he was funding programs about seed production, aquaculture, something related to biotechnology. And you all know that phase, when everybody became biotechnologists without having any training in biotechnology. But now, of course, people are attained, skills and knowledge. I think biotechnology is definitely a sunrise sector, especially. And we have done tremendous work in the pharma industry, health industry, animal biotechnology, agriculture biotechnology, and human resource development of innovation mentioned. The biotechnology, the skill development, so many programs in private sector, government sector. There are about 5,000 to 10,000, I don't know what is the number now, biotech industries in this country. And more than 5,000 startups. And many in the agri sector, in the medical sector. You all know that Biocon, started in Biocon, the first biotech industry. And then this is gaining momentum. So I have shared this, with you, the achievement in the last 5 years. And but then when it comes to aquatic biotechnology, our ICR Institute, universities, the institutes now have done a lot of work during the past several years. I mean, they have really become the centers of excellence, whether it is in agriculture biotechnology, from health to health, to genetics, different programs, molecular diagnostics, nutrition, genomics, the marine bioprospecting, from the marine organisms, bioremediation, biodiversity management. So rapid progress in India, or maybe whether you have programs on sequencing, whole genome sequencing. We all learned from Dr. Raj that they're working on human genome project, and so this has now been done in several species in fisheries also. So when we are talking about the blue transformation, blue economy, blue biotechnology, what is needed for future? I think that's what this conference has to address. See, the novel technologies, the novel approaches, first is to a kind of a sustainable management of resources. What biotechnology is required for sustainable management of resources? Disease resistance, improve feed, efficiency, mitigate climate change. These are the areas probably where biotechnology is play a major role in the fisheries sector. Should we not develop programs, breeding programs for heat tolerant species, disease resistant species? There's a requirement for future. So, DBT, I wanted to tell you that DBT also proposed to establish a National Institute of Marine Biotechnology. I know Dr. George used to share with us, and they had organized a National Institute of Animal Biotechnology in Hyderabad. And this National Institute of Marine Biotechnology was also discussed. Maybe a blueprint was prepared, but somehow this did not materialize. And then gene editing, is another excitement now. So everybody's talking about GMO, gene editing. And but GMO in case of fisheries has failed to take off. The transgenic fish, the students must be knowing the first transgenic fish, the salmon, aqua, adventist, which was produced 30 years back. 30 years back, it was produced and released in 2016 in Canada and 2021 in the US. But still it's not there. So regulations, along with the GM research, regulation part is also important. I think DBT has come out with a number of regulations in recent years now. I think DBT has come out with 2 regulations you all must be knowing. If the students are not aware, they should read about the DBT website. But what are these 2 new DBT regulations? And we also know that the use of AI and machine learning, that breeding, bioprinting and tissue engineering, they're all new excitements. And Goswami is already working on in vitro lab meat, tissue engineering. It's an excitement, global excitement. So there are several areas, probably several areas that we have made a headway. We have become, in fact, leaders, but there are several areas probably we have to still venture into. And as you all know, this kind of conference provides a place for making friendship, making partnership, and also to develop national programs, to bring more visibility to this institute. And I'm sure next time when we come, maybe to host this conference, maybe after a couple of years, we'll have a different auditorium. This is still, so I hope that this will have expansion in terms of physical facilities also. I know the laboratories and the kind of accreditation of the lab accreditation, has got this all new developments. Friends, the world is looking at India. The whole world is looking at India for everything now. And biotechnology is not exception. So to bring a real revolution in biotechnology, global revolution, India has to play a very important role. And I'm sure that you will come out with a roadmap, a strategy document for fisheries biotechnology. What kind of research you need nationally, what kind of research this institute needs independently, what kind of research you need in a multidisciplinary mode, what kind of research you need in a partnership mode, and what kind of global partners you will need for fisheries biotechnology to go to the high level. And the new knowledge, I think the insights and opportunity for partnership and the academic friendship, which you can develop, will definitely stimulate the younger mind here to take this sector forward. Thank you for inviting me, Dr. Vishankarji, and thank you all for coming and participating in this. And we look forward to your inputs to make this sector strong. Thank you very much.
Gratitude to you, sir, for your impactful insights, which are a guiding force in steering the fisheries fraternity through numerous challenges and helping us identify solutions with high accuracy and precision. Partnering with eminent scientists from higher organizations such as the Indian Institute of Technology, from where we have here today Dr. Sanjeeva Srivastava, we welcome you, sir, to this gathering. As the premier national fisheries university dedicated to fisheries research, education, and extension, ICAR-CIFE has been playing a vital role in nurturing and shaping the careers of its scientists, senior technical faculty, and students by bringing out their best original compositions, ensuring scientific excellence and professional growth across multiple dimensions. So, I humbly take this opportunity of requesting our Honorable Chief Guest, Dr. Raghavendra Bhatta, sir, and our esteemed guest of honor, Dr. W.S. Lakra, sir, to kindly do the honors of releasing 6 significant publications from ICAR-CIFE Mumbai. The first to be released is the Book of Abstracts of this International Conference of Fisheries Biotechnology, an ICAR publication. The next to be released is the divisional profile of the Division of Fish Genetics and Biotechnology, ICAR-CIFE Mumbai. The next 2 publications due to be released are technical bulletins. The first one, titled Advanced Techniques in Cell Culture, authored by Dr. Mukunda Goswami, Head, Fish Genetics and Biotechnology Division, and the second technical bulletin, titled Proteomic Expression Tools in Fisheries, co-authored by Dr. Mukunda Goswami and Dr. Sanjeeva Srivastava, Professor, Indian Institute of Technology, Bombay. I would request Dr. Sanjeev Srivastava to kindly share the stage. The next to be released is a manual entitled Art for Fingerlings, a beginner's manual for fisheries researchers, authored by Ramesh Roshan and Dr. Srinivas Jagirdar. I request Dr. Srinivas Jagirdar to kindly share the stage. The next publication to be released is a book of a different taste. It is a book titled Logo Lingos, authored by Dr. Dasari Bhumaya, who is a celebration of creativity, purpose, and meaningful design, having designed logos for prestigious ICAR institutions along with affiliations and special events. I request Dr. Dasari Bhumaya to kindly join the guests on the stage. I would like to tell the august gathering that Dr. Bhumaya has been called the logo man of ICAR. I now take this opportunity to request our Honorable Director and Vice Chancellor, Dr. Ravi Shankar CN, to kindly felicitate our esteemed chief guest and distinguished guest of honor with customary Shrifal shawl and a special memento. Our chief guest, Dr. Raghavendra Bhatta. I request Sir to kindly felicitate our guest of honor, Dr. W.S. Lakra, with the customary Shrifal shawl and memento. Thank you, sir, for doing the honors. It is our great honor and privilege to introduce Dr. Raghavendra Bhatta, Deputy Director General, Animal Science, ICAR, as our distinguished chief guest today, who is an eminent scientist par excellence with many feathers in his cap. As a renowned expert in the field of animal science and biotechnology, with an exceptional career dedicated to advancing scientific research, sustainable innovations in animal husbandry, he has been in the forefront of steering initiatives that have bridged the gap between cutting-edge biotechnology and practical applications to enhance food security, animal health, and productivity across India. His noteworthy contributions to the development of sustainable and innovative approaches in the realm of animal genomics and biotechnology have been exemplary in promoting interdisciplinary collaboration to address the global challenges in agriculture, earning him global repute. Under his leadership, the field of animal science has seen significant strides in research, policy development, and implementation of programs that have positively impacted rural communities and the agricultural ecosystem. Dr. Bhatta's vision of integrating biotechnology with animal science continues to shape the future of innovation in animal sciences in India, and his presence will undoubtedly inspire and guide us towards achieving greater scientific breakthroughs for the advancement of biotechnology and its applications in the realm of agriculture and animal sciences. We are uberly proud today to have you, sir, in our CIFE Parivar, and I'm truly honored to request you to kindly grace this podium and share your words of wisdom.
Good morning, esteemed directors, and Vice Chancellor of City Mumbai, and my close friend Dr. Ravi Shankar, respected Dr. Lakasar, former Director of City Mumbai, and Secretary Nas, Dr. Mukunda Goswami, Head of the Division and the Organizing Secretary, invited seniors, Dr. George John, Dr. Nana Sar, Dr. K.K. Vijayan, another good friend of mine, Dr. Wilson George, Director CMPRI, Dr. Girish, esteemed colleague from NRC, Nikhil, invited seniors, all the participants, students. At the outset, let me thank Dr. Ravi Shankar for this invitation. I don't know whether the madam with the biodata is, I think it is something, cut and paste, because this is what she has written. It's not really, it doesn't belong to me, because I'm not a biotechnologist, let me honestly with you. So basically, I'm a veterinarian, but you must be wondering how come Dr. J.K. Jena is not here. This is my second visit to this institute in a very, this span of very short time. Thanks to Dr. Ravi Shankar ji that he asked me to come for the fish swap, although I don't take fish. But I had a very nice time, visiting all the stalls and interacting with the students, and there are also some options for some vegetarian food, so I really enjoyed that. And this is the second time, when anytime the Shankar invites me, it's difficult for me to say no. So whether it is Parliament session or any session, I said, let me come to Mumbai again. So thanks a lot for the invite. And let me congratulate the CK family for organizing this third international conference on Fisheries Biotechnology for 2 days, 17th and 18th. I was just going through, there are about 225 participants. There are also international participants from Japan, from Singapore, Netherlands, France, UK, Canada, and US. Dr. Ravi Shankar did mention that, like we are not working in silos. It is interdisciplinary research work. So as a testament, I am here for a fishery biotechnology, a person from the animal science division. There are certain very commonalities, like when you talk about the biotechnology, because generally we don't say that biotechnology is a specific discipline. It's an amalgamation of many things, biochemistry, sometimes the nutrition, physiology, and genetics, and everything together. I think that way, like I thought, we're running late, as per the schedule, so I'll just take about 10 minutes. Friends, all of you know that, COVID has taught us a great lesson that is specifically with regard to the health and the immunity. And as it been mentioned that we are talking about now 2030 and 2047, the Vikasit Bharat. And we also have a target that if you look at the livestock meat production around 230, we are targeting around 600 million tons. And same is the case with the eggs as well as the meat. The one important lesson what the COVID, specifically has taught is the health. So in that, earlier there is a saying that, people used to say that when character is lost, everything is lost, when health is lost, and something is lost, and money is lost, something is lost. Now the total concept has changed, like we don't talk much about the character, but health is one thing, we always talk that when health is lost, now everything is lost. So in that, definitely the animal proteins, especially the egg, the meat and the fish, plays a very, very crucial role. And now we started talking about the smart proteins and as a function force. And biotechnology can offer a very, very powerful for the sustainable aquaculture because let me just say few of these areas which are more or less common to both fisheries as well as animal science. For example, we talk about genetic improvement, we also talking about genetic improvement. We're also talking about the disease control because of the climate change, the emerging and the re-emerging diseases. We are also working on more or the same line. Environment management, we're also talking about that. We have a collaborative project with CFI Kochi, with the nutraceuticals, trying to reduce the antimicrobial emission. Started with CFI Kochi. Now we already have 2 patents. We published some very good articles. Hopefully that in future we'll be able to make it as a part of the livestock ingredient. We are also talking about the biodiversity conservation. The same thing in livestock, we talk about zero ND because unlike in fishery, I don't know whether you have a fish which is recognized as a non-descriptive, but in livestock we have a lot of non-descriptive animals. We say more than 50% of them are non-descriptive because we have the in livestock census, which will be coming in another 2 months. And most of this, some of the livestock, suddenly we look at it, say it's indigenous or it is ND. There's a very popular terminology called ND, with the non-descriptive. So we have taken that pledge that by around 2030 and 2035 that we will reduce this to less than 5%. We are meeting all the efforts. Now coming to some of the areas where the genetic this biotechnology can really play a very critical role. One is the genetic improvement and reproduction control. Something like a selective breeding and the genetic engineering. We are all talking about the Cas9. We also do almost the same thing. We do the cloning. Then recently we have done the knockout of the beta galactose where globally somebody who's not very comfortable with the we have the lactose allergy. That people are not able to take. I think similar thing is going on in case of the fisheries. There's somebody's allergic to some of these. I think we must be working on those lines and show that fish allergy. I think I heard that it's very common, like as soon as something like allergy to brinjal. Some people are highly allergic to brinjal. If you take it in 15, 20 minutes to start with this. I think there's one thing, people have started working on that. And the biosecurity and the disease control. So this is also we are working, like everybody talks about the climate change. As we know that, per degree rise in the temperature, the sea level moves the rise at least about 30 centimeters. And the expected 2070 is going to be very true, and 2.5 degree centigrade. So roughly we'll have around 25 centimeters rise in the sea level in most of our coastal areas, hopefully not Kerala, but Maldives, Mauritius, and Lakshadweep, and part of the West Bengal. I think people who have seen that documentary on inconvenient truth by the Algo. I think it's very apparent that we are working on the climate change aspect as well, especially with regard to the methane emission, because I happened to listen to Dr. Jena Sarab, and he did mention that there's some work as far as the GHG emission from even the fisheries, which is I think very less, of course. Almost 30-35% blame comes to livestock. So we are a huge contributor. Not only that, because of course we deliver a huge number. We are almost about 500 million livestock, 300 for mostly the cattle and buffalo. So obviously we are contributing a bit on that. Now we are also working on the vaccines and the diagnostics, especially with regard to the emerging and re-emerging diseases. What we expect that if there's a climate change and there's a some of the glacier that will be melting, and we really don't know what is there underneath. So something which the disease must have happened about 500 or 600 years back. So most of them will be coming to the mainland. There's a work that's been carried out by the Nishad Bhopal that they collected some of these samples, especially from the Arctic area. What I've seen is some of these virus that has been identified in those dead birds which are underneath. They already there in West Bengal, so because of the migration. So we are also looking at all these, some of the pathogens which, because we are seeing that LSD suddenly all of a sudden they started skin disease because when we have the vaccination, I think similarly, I think same thing with the regard to fishery and aquaculture. Environmental management and bioremediation, I think getting rid of the pollutants, we are also working on that particular area, especially in regard to the algal biotechnology. This is specifically what I was talking about, which CFR is working. They are working on some of these seaweeds. After extracting the nutraceutical, it has got a very, very powerful anti-metallurgic compounds, especially with regard to reducing intermediate emission in case of rumen layer. And lastly is the biodiversity conservation, which is also very, very important. We always talk about the dilution of the geoplasm. And the other important thing is water. I think everybody says if at all there's going to be enough world water, it's not because of the oil, it's because of the water crisis. Here. So we're all more or less, I think we are working on similar lines and only the species is different. We are working on the livestock and you are working with the fishes. Otherwise, I think it's a strong possibility for collaboration. As you mentioned that, not working in the silos. Now, like City Institute, they're talking about one city. We also talk about one ICR. So, especially with regard to collaboration, common areas of mutual interest, that can be strong collaboration between the fisheries and the animal science institutions. And definitely, CAP is doing a great job in collaboration to the good words. Dr. Ravi has mentioned that yes, definitely. Dr. Ravi Shankar has done great work at CFE Cochin for a very long time. And after that, whatever the period he had got, I think I personally feel here that in a lot of transformation as far as the CPI Mumbai is concerned. Your whole team and thanks for this opportunity for inviting me over here. And I'm sure that what will the deliberations you do and what will the recommendations will be sent will be helpful for taking some of the policy decisions at the ICF headquarters level. Because very soon we will be having our new PFC. Hardly a few months up. I think maybe we can have some of those projects where animal science and fishery can work together, have a common protocol and common program, something like. Of course, we have, that this one on antimicrobial resistance, where AMR project, both fisheries and animal science, we are doing together. I think there are also common areas where you can work together. So, with this, let me close here. I wish you all have very comfortable stay in Mumbai and stay comfortable weather as well, not that hot as compared to peak April and May. And I wish this conference is a grand success. Thanks to Dr. Mukund again for all the arrangement. Have a nice stay over here. Jai Bharat. Thanks so much.
It was a real pleasure listening to you, sir. We sincerely hope to imbibe your qualities coupled with great voice as we stand on the precipice of this momentous occasion, taking your advice to help us navigate our way into more productive tomorrows. Finally, I'm happy to call upon our Joint Director, Dr. N.P. Sahu, sir, to present the formal vote of thanks.
Very good morning, all of you. All dignitary present here. This is the end of the annual program. So, here we are privileged to extend the vote of thanks to all the dignitary present here, especially the chief guest of this program, Dr. Raghavendra, the DDG, Animal Science of ICR. Though we got a opportunity 1 month, maybe 26 January, to be here as the chief guest in our fish festival, as he told that being a vegetarian, he also equally enjoyed or more than enjoyed beneficiarian, the Peace Festival. And your inspiration speech motivated all the people there to accept as a leader. I personally believe that true leader is not due to position and power. True leader is due to the wisdom and your inspiring power, inspiring to others. That is the thing that we have learned from you. Thank you very much for your precious presence here. Even if there is no time during mountain, that is financial end, still then you are here. Only because there is another bonding between Dr. Ravi Shankar and Dr. Raghavendra Bhatt. You always mention my friend, but this is a very rare example of the friendship. I have never seen anywhere. So this is made a compulsion to come here without saying no to Dr. Ravi Shankar. Thank you very much, sir, for present here and in this special occasion. And Dr. Our guest of honor, Dr. Lakhan, as he told, when I joined, he had a special lab, biotechnology lab. At the time, there is a very rudimentary. But at that time, you have laid the foundation stone of biotechnology lab. But you had a dream that you had that advanced research. So today you see that this conference is on advanced biotechnology. I think that your dream come true. You are the most befitting person to be the best honor in this function today. So thank you very much for your gracious presence and add value to this function. Dr. Ravi Shankar, that is a man of energy and commitment. And you know that the last, as we know that he is going to be superannuated next month, that is on April. So by default, any program coming, it will be the last program of his official career. So we are saying all the things last 2-3 months. Today also will say that this is going to be our last that is conference at CIP. But I have seen that the time of last 15 days we have so many programs. 2 days, last Friday we have completed our RSC. Before that, that is 1 day before, that is our convocation. Then 1 month or 15 days before, that is our peace one. 3 important functions of high magnitude. Still then, this program was scheduled and it was successfully completed. Thank you very much for your commitment and the energy and guidance given to all. Thank you very much, sir. Then Dr. Mukunda, the coordinator, I can say that he has organized these functions today, that the total hall is full. So I must thank Dr. Mukunda for his organizing secretary, along with him, Dr. Jagirdar, Nagpure, and all the staff of Fisheries Biotechnology divisions. And I know that is he was working behind the scene, but today only he has come to the front of the scene. So he has done nicely everything, something is there, but you know that we have committed, we have conducted this type of program in miniature form. We have conducted for aquaculture, conducted for nutrition, conducted for pathology, conducted for environment. And it was long due for him, and today it is completed. Only difference is that during the previous program, we invited the industry people. There is a close interaction between the industry and the students. It is throughout the day, 1 day program. Here, industry are not there, sponsors are there. So it's a different format, but definitely 2 days we have an intense discussion. As Dr. Lakra told, that the advanced biotechnology that we will discuss, it has been incorporated to almost every division. All are working on biotechnology, except in social science. Still, then they call it the molecular extension. So I congratulate all the scientists present here for doing research, extraordinary research in the biotechnology. In the coming years, it will be more fruitful. Besides this, the presence of the dignitary here, I can see the professors from IIS, professor from IIT, Dr. Vinson, Dr. Patil, Dr. Pidas, Dr. Kanta Mahapatra, Dr. Vijay, Dr. Ninabe, Dr. Puneya, Dr. Jones, and more, Dr. Das, all are present here. So I congratulate and thanks all the persons who are working behind these functions and to make it a grand success. I cannot take more time because the online, the speakers are waiting. So they are having a late night today, just now. So I also thanks all the speakers joined online. At the last, I congratulate all of you to make this function a grand success. Thank you all.
And I take this opportunity of thanking Dr. N.P. Sahu for presenting the vote of thanks. At the end of this inaugural program, we request all the August gathering to stand up in honor of the nation for the national anthem. जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्छल जलधि तरंगा तब शुभ नामे जागे तब शुभ आशीष मागे गाहे तब जय गाथा जनगण मंगल नायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे. At the outset, I request all of you to continue sitting in the auditorium for the keynote lecture by Professor Rex A. Dunham, who is going to join us from Auburn University, USA.
संजय वो गिरा के उसको ये कर देना हाँ स्टेज वैसा रहेगा उसको गिराइए ना स्क्रीन गिराइए ना नहीं वही ऑन था ऊपर में ऑन होगा
Thank you so much. Now we are going to start one online keynote talk that will be delivered by Professor Rex. Already he is right. So I request Dr. W.S. Lakra sir to chair the session. Sir, I request you, Dr. Lakra sir, I request you to chair the session, sir.
माइक सुनिए माइक नहीं पोडियम क्यों लाएंगे माइक ये इससे हो जाएगा इससे हो जाएगा सर कैसे I'll move once the online is finished then I will move this one सर को इसमें मेल लगा हाँ माइक संजय कनेक्ट किया हुआ है सब
All right, please take your seats. We are starting with the first keynote lecture by Professor Rex Dunham.
ये कैमरा किधर करेगा उधर करेगा
University.
ये तो पूरा खाली दिख रहा है
Let me just introduce Professor Dunham, who is a professor and a co-cultural geneticist at the Auburn University, USA. And
नीचे
he's a very renowned, geneticist and agricultural geneticist. The past president of the International Association of Genetics and Agriculture. We all know that that international conference now organized after every 3 years or 4 years. And I've been a part of this, many of these conferences, and I know I have met Dr. Rex during those conferences. And Dr. Rex has led research across 18 countries. You know, the expertise, and he has guided more than 50 MS and 45 PhD students from 19 countries. Over his more than 500 scholarly, works, and has more than 12,000 citations. And interestingly, the more interesting achievements of Dr. Rex Benham is the first to demonstrate genetic selection in channel catfish. He pioneered genetically improved fish release in the US. He achieved the first transgenic fish and CRISPR-based genetic modifications in aquaculture. They transformed the US catfish industry into a majority hybrid catfish sector. And then established the Southern Eastern Fleet Genetics Cooperative to tackle genetic management of catfish. So these are some of the key achievements for the Rex Benham. I think Dr. Benham, are you there online?
Yes, hello.
Okay. Okay, so it's a great privilege, honor to have you, one of the keynote speaker for this international conference on fisheries biotechnology. Thank you very much for agreeing to deliver this important lecture. And we have the here hall full of all senior colleagues, senior scientists, the young scientists, the science leaders from this country, the students, and several participants who are looking forward to your to listen to your enlightening lecture on genetic, genomics, and biotechnology for aquaculture enhancement, connecting the past, present, and future. Over to you, Dr. Rex Benham.
Thank you very much. It's my pleasure to be with you today. And as indicated, we'll be talking about the past, present, and future of genetics, genomics, and biotechnology for aquaculture enhancement. Okay, so fasten your seatbelts because we're going to fly. We're going to cover a lot of ground today. First of all, don't forget the basics. Take advantage of both the old and the new. They are both valuable. So the use of established, best-performing domestic strains is the first step in a genetic improvement program and the mechanism to make the most rapid initial progress in genetic improvement. So and not only is that the first step, but strains and families have a very large effect on the success of all genetic enhancement programs. So, for example, if we start selection with a poor strain, we do the selection, we will come up with a better fish. But if we start with a good strain and select, then we will eventually have the best fish. So let's take a look at our different genetic enhancement programs to improve body weight first. And traditional approaches such as selection as well as modern approaches is marker-assisted selection, triploidy when we're talking about older fish and large size, triploidy in shellfish, and genetic monosexing are all consistently successful. However, with gene editing and especially genetic engineering, we can accomplish the most dramatic increases in growth rate. Now, interspecific hybridization almost never results in an improved fish for aquaculture, but there are a few notable exceptions. One in the US, our hybrid, interspecific hybrid between channel catfish, Ictalurus punctatus, and blue catfish, Ictalurus furcatus. This fish shows heterosis for a large suite of traits, and when you look at overall genetic improvement for many different traits, it really is one of the best examples of aquaculture genetic enhancement ever. Now, there are also a few very good examples where interspecific backcrossing resulted in an improved aquatic organism. And one of these is a backcross between the Peruvian scallop and the bay scallop, in which several growth and size factors are improved. And in regards to shellfish, of course, triploidy probably has more impact than any other genetic enhancement program. I don't know if you can see the arrow But this is some of Ximing Guo's work. So in 2005, we can see that the triploid oysters are growing much better than diploid, but the performance of tetraploids is very poor. And what limited success we've had with fish, we see the same thing where tetraploids are very poor performers. But they've instituted this program of systematic interploidy breeding, and now, 11 years later, if you look on the far right-hand up side of the program of the slide, you can see that the tetraploids, their growth now is even double that of triploids using this ploidy selection program. So in the case of disease resistance, traditional approaches such as selection, marker-assisted selection, and now genomic selection, as well as interspecific hybridization, all are consistently successful. However, once again, the transgenic approach gives the most dramatic results. So in this example, we're putting the antimicrobial peptide genes from alligator, cathelicidin, and also from moth, the sacropin AMP gene into channel catfish. And we see large, the data that you see is when these different transgenic types are compared to the control, and they are improved for columnaris resistance. But also, they're improved for resistance to Edwardsiella ictaluri, Aeromonas, parasites, and even to some extent improved ick performance, resistance to ick. In this example with the rainbow trout, when they put the sacropin transgene in, it increases bacterial disease resistance, but it also has antiviral properties, and these fish are more resistant to salmonid viruses. Now, I'm using this as the sole example, but when we integrate a transgene, and in this case, sacropin, we see differential gene expression for a whole suite of the native genes. So the transgene interacts obviously with the native genome, and we see a lot of differential gene expression. So overall, I don't have time to go over all the examples, but there are numerous examples where antimicrobial peptide genes inserted into fish resulted in outstanding improvement in growth resistance. There's only a few examples in regards to gene editing for improving disease resistance, and they're not nearly as dramatic. I can't give you any details, but maybe in the near future, there'll be the first very dramatic increases in disease resistance coming out of some work in your home country, India. Now, in sliding back to traditional selection again, there are some dramatic cases in increasing survival. So we see in the United States about 5 generations ago, you have heavy mortality in oyster populations. But after 5 generations of selection, we see a very dramatic result, about a 9-fold increase in survival. So there are cases, seems like shellfish, we have a lot of unusual but very successful examples of genetic improvement. Now, if we focus on reproduction, there's not as many genetic enhancement programs that will result in improved reproduction, but 2 of them, selection and intraspecific crossbreeding, can fairly consistently improve reproduction. Of course, marker-assisted selection is fairly new. There's many years of data actually now. And as you know, that's selection based on a DNA marker that's associated with performance. Now, there are 2 problems with this. One is we tend to focus on these new technologies, but if the heritability is already high, marker-assisted selection is no more successful than traditional selection. Also, usually these markers are strain specific, so if we change strains, we have to start the work over to identify the correct marker in order to have success. But there are some very wonderful examples here. In this case, they learned that a single gene was responsible for resistance to lymphocytis disease in Japanese flounder, and through marker-assisted selection, they were able to develop a population that had 100% resistance, and this is commercialized in Japan. Now, again, usually feed conversion efficiency is a low heritability trait. So in this case, with rainbow trout, they're able to triple the rate of genetic gain by using MAS. And in another rainbow trout example, in one generation of marker-assisted selection for a mitochondrial DNA marker, they were able to improve growth rate by 26%, which it took 6 generations of traditional selection to achieve the same result. So real advantage there for MAS, but not always. When we look at traditional selection versus MAS for bacterial cold water disease, the success is similar. So sliding into our modern genomic selection, of course, in this that are associated with the performance, some of the same problems. Usually, these SNPs are strain specific. So if we change strains or breeds, we have to retrain, do the retraining, and there's a lot of background work and expense every time we switch populations. And again, if it's the main benefit here is to improve low heritability traits. And in this case, continuing with that bacterial cold water disease in trout, the genomic selection, the marker-assisted selection, and the traditional selection, in this particular case, give you the same amount of improvement. And obviously, traditional selection in that case is more economical. In Zhao et al. In 2021, there's not many real response to genomic selection experiments, but in this case, they're selecting for resistance to a parasite in the yellow croaker. The results are outstanding. The genomic selected line had 60% survival versus 10% for the control when there was a challenge. And if they ran the challenge long enough, all the controls died, and 41% of the genomically selected yellow croakers lived. However, the heritability for this trait is very high. We don't know. They did not try the traditional selection, so we don't know if the genomic selection was actually better or not. But again, another really outstanding example coming from oysters. In this case, they have 2 generations of genomic selection for survival when challenged with dermo, and 2 generations resulted in a 70% increase in survival, and this is about 3 times, a good 2 to 3 times faster rate of genetic gain compared to traditional selection. So let's focus on genetic engineering. We call it a modern technology, but it's been ongoing in fish now for 40 years. The biggest set of data we have is many different labs and working with many different fish have transferred growth hormone gene into fish, and it's almost always successful. But depending upon the fish, the transgene, etc., the fish grow 30, 40, sometimes double, sometimes 5 times, 10 times, and in 1 or 2 cases, actually 30 times faster than normal. Truly dramatic. So in this case, those fish are growing faster because of a combination of increased feed consumption and better feed conversion efficiency. Now, actually, if we look at all genetic enhancement programs with aquatic organisms, especially fish, that improved growth rate, the faster growing genotype is growing faster because of both of these factors. However, the relative importance of those two can vary from case to case. Now, in this case, this is growth hormone transgenic tilapia, and as all of you know, the control males are growing about 40% faster than the control females. But in the case of the transgenics, there's no difference in the growth of the males and females. So we also have two other cases, one case in channel catfish, one case in salmon, where the female seems to respond much better to the growth hormone transgenesis compared to the male. Okay, and across the board in GH transgenic fish, protein's going to be elevated 10 to 20% in the flesh with a corresponding decrease in fat. So we have many different pleiotropic effects associated with the transgene insertion. In this case, for both channel catfish and Atlantic salmon, growth hormone transgenic fish, there is a change in the muscle ultrastructure. So in both cases, there are a greater number of mitochondria and glycogen globules, fewer fat globules, greater numbers of muscle fibers, but the same size of muscle fibers in the transgenics compared to the controls. The growth hormone transgenesis also changes body shape, which slows down the swimming in salmon, and in the case of common carp, a positive effect, it increases the dress-out percentage. In another example with GH transgenic common carp, again, in China, they found that the transgenics had a better dress-out percentage, but that was because of reduced reproduction and reduced gonad size in the transgenic fish at harvest. In the case of channel catfish, growth hormone, when you insert growth hormone transgenes, you get better survival in sub-zero temperatures and also better survival in high salinity environments. In the case of disease resistance, we look at all the examples, the results are quite variable, and that is illustrated in this single study where transgenic growth hormone salmon sometimes had higher survival, sometimes lower, sometimes the same survival when challenged with Vibrio. And so this appeared to be primarily a family effect. So we can't, when we look at modern molecular genetics, the traditional quantitative genetics is still important. In this case, it probably wasn't a very good idea, but the Canadians were taking antifreeze protein gene from winter flounder and transferring it to Atlantic salmon to try to make it so they could survive in the Arctic environment. Now, they did get expression, but no biological effect, no change in cold tolerance. They also did this work in goldfish, and it did not change the lower temperature that they could survive, but in their natural temperature range, it did improve survival in the lower end of their natural temperature range. So it did have a biological effect there. In regards to transgenesis, we need to do more work on traits that consumers are interested in. That would probably increase public acceptance of this technology. And one option is to try to improve the omega-3 fatty acid levels in fish, which of course, as you know, the omega-3 fatty acids have many, many important human health aspects to them. And also, not only are the omega-3 fatty acid levels important, but also the ratio of the N6 to N3 fatty acids. With if the ratio is favoring the N3 fatty acid, that is a positive health aspect. This is an example from India from, let me see how long ago, 30 years ago. And what we see is as the people had higher income, it actually allowed them to purchase and consume food that was more unhealthy. So as Indians gained income in their diet, there was a higher N6 to N3 fatty acid ratio, and that was strongly correlated with an increase in diabetes in rich people compared to poor people. So in the case of fish, tilapia and channel catfish, they're very low in omega-3 fatty acids, but fish like tuna and salmon are high. So there's been considerable amount of work transferring omega-3 fatty acid biosynthesis transgenes to attempt to increase omega-3 fatty acids in fish. And the overall result increase the omega-3 fatty acids about 20 to 100%. And we also get a corresponding improvement in the omega-3 to omega-6 fatty acid ratio. Now, in this case, Pang et al. Transferred fat 1 and fat 2 into zebrafish and obtained a 2 to 3-fold increase in N3 fatty acids. We attempted this in channel catfish, had positive results, but not as good as in the zebrafish, 25 to 50% increase in N3 fatty acids. Now, the Chinese, they seem to do a lot of magical things. In this case, they randomly cut DNA from shrimp and randomly inserted it into common carp, so they weren't sure exactly what genes were being transferred, but a sample of these fish had improved flavor and improved amino acid and nutritional profiles. Now, transgenesis can also be used to produce biomedical proteins. In this old example here, the human coagulation factor 7 transgene produced that protein in transgenic zebrafish, walking catfish, and Nile tilapia eggs. The clotting factor protein produced by these transgenic individuals had the proper post-translational modification. The proteins could be collected from egg, serum, and probably other tissues as well, since it's a constitutive promoter. So the very first transgenic fish that were commercialized were actually glowfish, both Medaka and zebrafish in Taiwan and Singapore in the U.S., that were produced by transferring fluorescent protein genes from the jellyfish. And today in the aquarium trade, we see many different species and many different colors produced by this process. Now, in regards to a lot of the basic research that's done using green fluorescent protein gene, we really need to examine those results and think of them carefully because GFP reduces cardiac function and aerobic performance in these transgenic zebrafish. So we really need to make sure this research is meaningful. Let's talk about gene editing for a little bit, and I'll start with just a really fantastic result that we had with channel catfish, where we knocked out the melanocortin-4 receptor gene. By knocking out this one gene, we were able to sterilize the fish. It increases their growth, it increases their fatty acid levels, and it also increases disease resistance. One knockout, 4 traits improved. There's been several studies where in fish where the growth regulator myostatin has been knocked out, which of course in mammals causes the double muscling when we see this natural mutation. In the case of fish and channel, this slide is of myostatin gene edited channel catfish on the right, control on the left, and the mutated fish have about a 30 to 40% increase in muscle fiber number, but the size is not increased. Now, similar research with common carp, we have a slightly different result. You obtain both increased muscle fiber numbers and increased muscle fiber size. In this experiment, of course, carp have those pesky intramuscular bones. So Zhang et al. Knocked out SP7A and SP7B in order to eliminate these intramuscular bones. Now, the problem in this case is it caused numerous different changes in the various bone complexes, resulting in almost a subviable fish. In this second example, they knocked out BMP6, the bone morpholinose protein 6 in crucian carp, and in this case, they were able to eliminate or reduce the intramuscular bones and have a very viable, and in fact, they claim that it was a stronger fish. Now, this is an unusual gene editing result. In this case, in Medaka, they're knocking out tryptophan hydrolase 2 gene, and by doing that, they're not able to synthesize serotonin. The result was some very relaxed fish, so the behaviors change. They're less aggressive. So using gene editing to chill fish out. Now, we've been focusing on single genetic enhancement programs, but combining multiple genetic enhancement programs, just like you're baking a cake, working on your car or house, to get the best result, we need to do the same thing in aquatic organisms. Here's another oyster example. If they do a combination of crossbreeding and triploidy, you get a better result than crossbreeding alone or triploidy alone. Now, there's been several papers that looked at simultaneously using multiple genetic enhancement programs. Just a few examples here. Of course, gene knock-in, knock-out. In salmonids, they've done a combination of selection, polyploidy, and genetic monosexing. Crossbreeding and selection have been combined, selection and genetic engineering, etc. So looking into the future, we're not past, present, and future. I firmly believe that in the future, all of our best genotypes in aquaculture are going to be by simultaneously utilizing multiple genetic enhancement programs. So in the case of gene editing, and in particular genetic engineering, are we ever really going to see deep impact on aquaculture production and world hunger? I believe that it is coming. It may be slow, but it is coming. Of course, in gene editing, it's here. But transgenics is partially here, but it's going to take longer. So there are concerns about these two technologies, especially transgenics. Are these fish safe to eat? Are they safe for the environment? And the problem, what's causing these questions is we have a real problem with public education regarding biology, food production, genetics, and then, of course, all that adds up to educational problems regarding transgenics. So here's the type of public education that the general public throughout the world can easily understand, but it's false information. And to illustrate this problem that we have, this is actually some surveys that Thomas Hoban did in the 1990s to try to see if people really understood transgenic technology. So he asked the simple true-false question: ordinary or normal tomatoes do not contain genes, while genetically modified ones do. Now, the obvious answer is true, but even if you don't know, you have a 50 chance of answering correctly. 52% of Canadians could answer this correctly, only 45% of Americans. And my ancestry is primarily from the UK, so I have to admit it gives me a little bit of pleasure saying that only 32% of the French could answer this question. So in this case, here are major scientific organizations throughout the world, and they all did white papers. They all came to the conclusion that there is no logical reason that transgenic meat causes a food safety concern. Even the European Food Safety Authority concluded this, even though Europe and this organization are both highly anti-transgenic. But we do have to be careful. We have to be smart. There are exceptions. It is possible that if you transfer the gene that makes the protein of peanuts that elicits an allergic response, same thing for shrimp, corn, eggs, and a person ate that transgenic fish, they could have an allergic reaction. So we do have to look at things, obviously, scientifically and case by case. Now, in regards to environmental risk, the main fitness characteristics are reproduction, and they're interrelated: reproduction, foraging ability, predator avoidance, and swimming ability. There's been many studies done on transgenic fish looking at these factors in simulated natural environments. And there are some exceptions occasionally for one of these traits, but overall, the fitness of transgenic fish is reduced compared to wild type and wild fish, so you would expect them to be outcompeted and eliminated in the natural environment. And one example is in an aquaculture environment, we're spoon feeding the fish, and we see the growth enhancement. But there's more than one experiment that shows if they have to eat natural food, catch it themselves, there's no enhanced growth and maybe even slower and survival problems. So this is a genotype environment interaction. And I'm not going to spend time on it, but every genetic enhancement program we've talked about today, we need to be aware of the genotype environment interaction. Whether it is traditional selection, transgenics, or even in pure genomics and functional genomics work, the genotype environment interaction is important and can influence the results and whether we're coming up with something that's better for the farmer or not. So overall, the data indicates that the environmental risk of transgenic is quite low, but that's not good enough. Today, whether they're fit or not, with the current public opinion and regulatory environment, it's going to require confinement for application of transgenic fish. So how do we control these fish? How do we confine them? And this really is not just an issue for gene-edited and transgenic fish, as there are other controversial aquaculture genotypes. So physical confinement and RAS, etc., is not the answer, because we all know, we learned from Jurassic Park that there are mad scientists who will steal germplasm, they will steal dinosaur eggs, and spread them out throughout the world. So physical confinement is not the answer. Reproduction is. genetics is, sorry. So what options do we have to sterilize or reduce the reproduction of fish? Hybridization, polyploidy, genetic monosexing, they can all do the job or partially do the job, but they don't eliminate risk because all these programs require fertile diploid broodstock to be maintained. But we have great potential with both gene editing and with genetic engineering. So in the case of genetic engineering, genetics is the best approach for long-term total reproductive control. In this example, we've used a short hairpin RNAi transgene construct to knock out primordial germ cell genes in channel catfish. As you can see, this male has no testes. So actually a transgenic approach will work. So these are full siblings. The top fish has no gonads because of the PGC transgenic knockout, but by incubating the eggs in a solution that will turn off the promoter, we can have normal development and therefore have an aquaculture farm production system. But if the fish escape, they can't be exposed to the proper chemical and all the progeny will be sterile. Now, the problem with this is apparently it's important to have your gonads for normal growth and survival. So the transgenic individuals in this case from the PGC knockout, they grew 25% slower and they had 25% less survival compared to the controls. However, some of those transgenic individuals were as larger than the controls. So again, the old-fashioned approach, selection, may be one way to fix this problem. Of course, another option for total reproductive control so that the fish can only mate when the hatchery manager intervenes with hormone-induced spawning is by knocking out the genes from the HPG axis or perhaps other reproductive genes. Now, in this case, this is some work done by CAT with tilapia. If they knock out, they gene edit FSH receptor gene, these tilapia develop no ova at all. In our case, knocking out the HPG axis genes. On the top left, some of them will lay eggs, but you can see here the eggs are fungus. They're all dead. On the right-hand side, this is a full sibling. They received hormone therapy, hormone induction, and they spawn. And the embryos, we have normal reproduction. Now, in the fish world, as you all know, there's many different reproductive strategies. The genetic mechanisms for sex determination, etc., and spawning are tremendously variable in the teleost world. So this successful approach in channel catfish for most cases of HPG axis genes in Medaka and zebrafish, it doesn't work. You knock out those genes, and in most of those cases, they still have partial or full reproductive capacity. So if we're able to knock out these HPG genes to get total control of a transgenic fish, we need to be aware of potential pleiotropic effects. Just as in selection, we have to be aware of possible negative correlated responses. So if we knock out GnRH1, in the case of catfish, it almost always decreased growth rate. If we knocked out GnRH2, it almost always increased growth. In the case of knocking out LH, a variable, sometimes decrease, sometimes increase, sometimes no effect. Not as much data, but knockout of FSH tended to increase growth. So let's keep rolling into more modern genetic enhancement. So now that we have CRISPR-Cas9, we're able to do knock in, knock out. We can do targeted gene insertion or replacement. And in this case, we're going to knock in, knock out multiple genes, kill several birds with one stone. So in the case of catfish, we have transgenes, cecropin and cathelicidin, that we want to knock in to increase disease resistance. Different omega-3 fatty acid biosynthesis genes from salmon, rabbitfish, and C. elegans to increase omega-3 fatty acids. Our target loci for knockout are growth regulators, MC4R and myostatin, fertility genes such as MC4R and LH, and omega-3 fatty acid genes such as MC4R. So our goal is to, in one fell swoop, increase disease resistance, growth, fatty acid, omega-3 fatty acid levels, as well as sterilizing the fish. And we've had success. On the top two lines, we're knocking in cathelicidin at LH or randomly. We get about a doubling to tripling of disease resistance when we challenge with ESC. But then with columnaris, you see much greater dramatic results, greater increases in disease resistance compared to the control. And we see that over and over and over. ESC is a more difficult pathogen to control compared to columnaris. Now, the rest of the slide in the left-hand column, looking at ESC, so if we do a single gene knock-in or knock-in, knock-out, we get a 2 to 3-fold increase in disease resistance. But if we do a double knock-in, knock-out at growth regulating or fertility loci, we get 4 times increase in ESC resistance. So what effect did that have on body weight? If we just randomly integrate cathelicidin or cecropin, the fish appear to be healthier. They grow about 10% faster. But then if we knock out the growth regulators, either a single knockout or double knockout, about a 35 to 65% increase in body weight is obtained, as well as the increased disease resistance. So again, we need to be, what effect do the target sites, the direct and pleiotropic effects on the target sites when we do the gene replacement? Now, the knock-in, knockout is not always totally efficient. Sometimes we get on-target knock-in, sometimes off-target knock-in. Now, in the case of cathelicidin, where our target is LH, it doesn't, in regards to the disease resistance, it doesn't make any difference. The on-target and the off-target cathelicidin transgenics have the same disease resistance when we challenge with columnaris. And if we challenge with Edwardsiella ictaluri, same thing. They are both superior but equal to each other compared to the control. That's the yellow and orange lines versus the blue. Now, things don't always go exactly like we hypothesize. In this case, we are knocking the salmon elongase gene by knocking out the LH locus. The fish grew normally. In the case of the DHA levels, we increased those by 62%. And then total N3 fatty acids, a 41% increase. But we had a large number of these fish that showed an off-target deformity. Now, in this case, we're knocking out LH again, but it's in blue catfish. We're knocking in cathelicidin, and knockout in LH in this case had no effect at all on body shape and no deformities. So in this example, we're knocking in the common carp beta-actin promoter Masu salmon elongase gene again. but different targets. So there in where it says R and it's shaded in yellow, that's random integration. We see no effect on EPA and a 21% increase in EPA. And then on the far right, that is a targeted knock-in at the non-coding region at chromosome 1. We get the same results as in the random integration. Now then, to the left, we have our target is MC4R. We get a huge increase in EPA, 97%, a very good result for DHA, 33%. But if we do the knockout, the editing without the knock-in of the elongase, we get about the same result. Now on the far left, elongase in, LH out, EPA, nice result, 18% increase. DHA, our best improvement, 62% increase in that N3 fatty acid. So here we're looking at the pleiotropic effect on growth at different target sites if we knock in cathelicidin. On the far left, if we knock out at LH, anywhere from a negative 17 upwards to a positive 16 percent increase in growth rate, so quite variable. Same thing, but not as variable if we do random knock-in, anywhere from negative 9 to positive 6% increase in body weight. The fish that were integrated at non-coding region of chromosome 1, 11% increase. At MC4R, our growth regulator, we knock it out while putting in cathelicidin, 53% increase in growth rate. But the double knock-in, knockout with MC4R and LH only at 20%. So the LH knockout seems to modulate that increase in growth rate. Now the bottom line is the reproduction. So if we have these transgenic sterilized fish, can we restore the fertility? In this case, for these LH these LH knockouts, we learned that we need to use a combination of LHRH and HCG to restore fertility. So the transgenic fish and the wild type, they spawn, and their spawning rate is not different. The replication, though, is low. The knock-in, knockout fish are sterile, as expected. So we were able to restore fertility by using the LHRHA and the HCG, the right-hand side graph. However, their spawning rate and their fecundity is lower than the transgenic control and the wild type control. So we're going to have to do more research to improve the hormone therapy in order to restore the fertility and the reproduction to the same level as the non-sterilized fish. So I'd like to wrap up with a technology that's coming on that we may be able to use for further genetic enhancement in the future, and that's xenogenesis. It is a method of reproduction where successive generations differ from each other and no genetic material is transmitted from the parent to the offspring. A xenogen is an organism comprised of elements typically foreign to its species. If you're explaining this to lay people, it's analogous to human surrogacy, except the host is not carrying the baby of the donor. The host is carrying the gonads and gametes of the donor. So this procedure can be done with fresh tissue. It can be done with cultured cells. It can also be done with cryopreserved tissue, gonadal tissue, or stem cells. So why are we interested in this? Well, it's a hatchery tool we can use to improve fry production of difficult to spawn fish or fish with long generation intervals. My lab's interested in it as an alternative, more efficient way to make interspecific hybrids. It could improve transgenic technology. It's an alternative and faster technology to produce YY populations. It's a tool we could use to avoid inbreeding. It can accelerate all genetic and reproductive research. Um, okay, we've already covered those. It's a technique that we can use to expand endangered species. We can clone gonads of valuable individuals. And actually, because we can cryopreserve the entire nuclear genome and the mitochondrial DNA genome, as well as the cytoplasm, in contrast to sperm cryopreservation, if we have tissue or cryopreserved cells in the freezer, if we thought ahead and already had those samples there, we can actually resurrect extinct species, or more practically, extinct genetic lines by using xenogenesis. So the basics of how this works is we extract the stem cells, we make a sterile host, we inject those stem cells in the sterile host, they grow up to broodstock. In our case, we take a xenogenic channel catfish. He's producing blue catfish sperm. We mate it with a normal channel catfish female. The result is 100% interspecific hybrids by mating two channel catfish semi-naturally. Of course, we can also use that to produce pure blue catfish if we use the xenogenic female with the xenogenic male. And I really like this photo because this is a 3-month-old channel catfish. It's a triploid. There should be no gonadal development, but you can see there in the center, clear ovaries. Those are blue catfish ovaries growing, diploid blue catfish ovaries growing in that triploid channel catfish female. So some examples, triploid salmon have been used to produce pure rainbow trout fingerlings. Nile tilapia have been able to make Jundia catfish sperm. And in our case, we were able we mated two channel catfish together. On the right-hand side, we see channel catfish controls. On the left-hand side, you can see the morphological difference. Those are hybrid fingerlings produced by mating two channel catfish. And we've demonstrated that the reproductive ability of the normal channel catfish and the xenogenic catfish is the same in regards to spawning percentage, hatching percentage, and fry per kilo. So how we can usually use this in genomics and genetics is this is a technique that we could fix whatever genomic gain, or if we're doing interspecific hybridization, a reciprocal recurrent selection, breeding values, we can fix them. So what makes this, what allows this is that these stem cells are isogenic. They haven't gone through meiosis yet. So when you collect stem cells from a single individual, they're genetically identical. So we can make an entire population of cloned gonads and breeding values. So how this would work, we could have our very best, the best genetic blue catfish male ever, the ultimate channel catfish female for producing hybrids, or you can substitute in any species or combination that you want. So we harvest the stem cells from each line. We produce the xenogenic individuals. So we have a xenogenic channel catfish male with the testes and breeding value of the ultimate blue catfish. We mate him with a xenogenic channel catfish female with the gonads breeding value of the ultimate channel catfish, and we produce the ultimate hybrid progeny. Now, on the right-hand side, we don't need those xenogenic males, so we harvest the stem cells from them to repeat the process. And we can do this for 1,000 years, 1,000 generations. Same thing on the left-hand side. We don't need those xenogenic males, so we harvest their stem cells, and cyclically we keep repeating the process over and over without any change at all. The genetic performance of those progeny at the bottom of the figure, they aren't going to change forever. So that's it. Thank you. And we here we have the past. The last is a pictures from the past. Thank you very much.
Thank you so much, Kundaraj. It was very enlightening, and very comprehensive talk, you delivered on so many aspects you covered. Of course, the main focus was on the pandemic approach, and we covered both ornamental and the food fish, the market selection, and then boneless fish, so interesting, programs you are dealing with, omega-3, omega-6, and then one multi-billion-dollar question is about the public education, you said, to, create awareness of the public of all the GM food, and the potential of gene editing, and of course, the targeted gene replacement you mentioned, and finally, the gene. They're all very exciting areas. Some of them have already, established and transgenic produced, but some of them, like the uregenesis, still in the infancy. I think everybody has, loved your presentation. So rich and so informative, and with new knowledge and new initiatives, the global initiative. We would have taken one or two important questions from the audience, but then they are all running behind schedule. All right. And so we would like to profusely thank you. We would love to have you physically here maybe in near future, but thank you for agreeing to give this online presentation. And on behalf of the Institute, the Central Institute of Fisheries Education, and on behalf of the entire audience here, both physically and online, we once again congratulate you and thank you so much for sharing your thoughts.
You're very welcome. Thank you.
So, what is the announcement now?
Sir. Okay, thank you, Professor Dano. Actually, we met you long back in Bangkok, and then again, connecting with it's a great. And thank you so much.
Thank you so much.
You're very welcome. Thank you. It was a pleasure. Have a good meeting.
Thank you. Sir, we will break for a group photograph, then for high tea. Then after high tea, again we will be coming here. Another keynote speakers, 2 speakers will be here. So thank you so much. Thank you, sir, for sharing this important session. Thank you. Sir, reception. Just in front of this institute reception, already arrangement has been made. So we'll go here and have a photo. And we'll go for high tea. And then again we'll back here. Thank you, sir.
I close and keep. You have Wi-Fi, good Wi-Fi. I connected.
Yeah, Wi-Fi. I'll give you Wi-Fi.
So then I'll just.
Yeah, sure. Don't worry about it.
I connected.
No,
saying signal is very weak.
Yeah, because the ground floor. I'll give you Wi-Fi. मग तो उसको म्यूट कर रखे
दो एक घंटे
ये देखो सर सर सुनील
Actually, we are fortunate to have him here. He is a very quiet person. It's very difficult to get his time. Even if you go to IIT Bombay, hardly 5-10 minutes. He's so busy, and it is an internationally renowned scientist, and he is a product of Harvard group, university. And then currently he is a professor in IIT Bombay and specializes in proteomics, clinical research, infectious diseases, cancer research. And he did his PhD from University of Alberta, Canada, and postdoctoral fellowship at Harvard Medical School. The research focus on infectious disease. He worked a lot actually during pandemic time, also COVID time, the malaria, dengue, and COVID-19.
It is
And brain tumors
PPT file, na,
and
sir?
common cancers in
It's a
India,
PDF file, na, sir?
oral and cervical and breast
It's a
cancer.
PDF file, I think.
Proteomic database development and software innovations.
It's not PDF.
And he is a visiting professor in various prestigious institutes in the world, UCSF, USA, Nottingham Trent University, UK, Central South University,
Okay, so let me
China,
know. Let me know.
and a key achievement, he has 190 plus research publication, including his publication
now
in
it is okay?
like Nature, and then a similar kind of journals. So you can feel about this kind of work. And then 30 patents, 30 plus patents. So, and then mentor 26 PhD students. And leadership role in HUPO, that is a human proteomics system. And then Pi-Hub, recognized by Royal Society of Biology, Chemistry and Medicine. The impact globally recognized for advancing healthcare, diagnostics, and mentoring future scientists. And then whenever he organized international conference,
remotely, sir.
many of our students and faculty
easier
members
for you.
witness that. That's a very huge gathering, and he used to bring all international renowned scientists physically. So we are glad to have you, sir. Thank you so much for accepting our invitation.
Thank you, Dr. Goswami. And now I first my co-chair of the last. All right, welcome back everybody from the post-tea session, and it's really great honor to introduce our next speaker, who is one of the pioneers of DNA fingerprinting and genomics in India. Dr. Thangaraj and Dr. Lalji Singh, I think they were the backbone for starting the genomics and DNA fingerprinting in India. And I must say that I'd like to share a personal note on this, that I was a summer trainee student at CCMB Hyderabad several years ago. Of course, I never worked with Dr. Thangaraj, but my other summer trainees, they were always, praising about him, how precision you need in doing the DNA fingerprinting and genomics research. And of course, he has over the years shown the mentorship role and the leadership role in different institutes, from the CBFD to the INSTEM, and of course, the CCMB Hyderabad. So we are really, I think this entire gathering is very honored to have Dr. Thangaraj, who is going to talk to you about looking at genomics evolution, different health aspects. And he is going to give you a much bigger picture of looking at India from different genomic landscape, and I think that's going to be really a good perspective for all of us. So with this, let's welcome Dr. Thangaraj, who is a recipient of several national awards, especially the Bhatnagar Award and the JC Bose Fellowship, and so many excellence awards to his cap. So Dr. Thangaraj, we would like to welcome you and open for your session.
Thank you so much for the introduction. Thanks, Dr. Doctor. Also, thank Dr. Goswami for inviting me to talk to you. I told him we had a very small proportion of the work in collaboration with Dr. Lakra and Dr. Goswami several years ago in the area of fish, but I told him that I cannot talk back. It was 15-20 years back, but he said no problem to talk about human work which I have been doing for the last 3 decades. So although it's not relevant to fish, but I'm going to talk to about our own origin, and that way they are connected with what they are going to, or what I'm going to talk to you. So the title is Genomics in Evolution, particularly human evolution, our health and disease. So I'm going to connect you both evolution and genomics. First, let's see what is genomics in terms of the we all know genomics is a complete set of information present, genetic information present in a cell. But according to Francis Colley, who has involved in human genome sequence, he has put it in a simple way, which I read here. Genomics is a short manual with an incredibly detailed blueprint for building every cell, in this case, human invention. It is a history book, a narrative of the journey of our species through time. Genomics is a transformative textbook of medicine with insights that will give healthcare providers new powers to treat, prevent, and cure the disease. So in my talk, you will see both these aspects. Right? Now let's look at evolution. In fact, there's no genomic variation or genetic variation, there's no evolution. Whenever there's a change in our genome, it takes new features, that's where the evolution happens. I would like to show this particular slide where Jansky, one of the known evolutionary biologists, he has written an article in the journal called the American Biology. The title of that article is Nothing in Biology Makes Sense except in the light of evolution. So how true it is that you understand that without evolution, without reading evolution, you can't really understand many aspects, in this case, our origin, health, and diseases. So one of the interesting aspects, or one of the large number of questions which people asked in the last several hundreds of thousands of years, is how such a huge human diversity arose in India. We have 4,635 anthropologically well-defined population groups. How we all reached here? So these are some of the questions which people have asked. And it's well known that the modern human, originated in Africa about 160,000 years back. From there, how did they migrate and now inhabited all over the world? There are two hypotheses. One group of historian, archaeological anthropologists, geneticists, linguists, they believe that the modern human taken the northern land route because there's a land connection. But others did not believe on that because the climatic condition, even today in Europe, is not easy for the migration of the people about tens of thousands of years back. Therefore, they believed on the alternate hypothesis that the modern human might have taken the southern coastal route based on some of the archaeological evidences. And as I mentioned, we have been working on for the last 3 decades to understand this. I will give you selected studies which will connect the migration of the people. So we had opportunity to go and collect samples from the tribal populations of Andaman and Nicobar Islands several years ago. This is the Home here. There are 4 different tribal groups. Home Great Andaman Sentinels, Jawas. Sentinels are still in isolation, whereas others we were able to collect the sample. And we used complete mitochondrial genome. The last talk, the speaker spoke about mitochondria and mitochondrial genome. And we all inherit mitochondrial genome only from the mother. There's no contribution of paternal mitochondria. There are very rare cases, but always causes a disease that we inherit the paternal mitochondrial DNA because of the heteroplasm. We sequence the complete mitochondrial DNA, which is about 16,500 base pair. And based on the mutations, we propose that probably the Andaman is the first modern human who migrated out of Africa, taking southern coastal route and reached Andaman and Nicobar Islands about 65,000 years back. So this is the first genetic study. Of course, 20 years back, there's no other hypothesis that tells that no, this is not the right way. The first modern human migrated out of Africa. That still holds true. Subsequently, after human genome was sequenced, there were 1 million genetic markers from autosome, the 46 chromosomes are identified. We are using that information try to understand the Indian population history. So we used the 1 million genetic markers. So in this study, in fact, we have reconstructed the Indian population history. So what we did was we used selected population groups all over India and used 1 million SNPs and shown that in the prehistoric India, there were two founding populations. One called them as ancestral South Indian, who probably part of the early human migration. And the migration happened, some groups stayed in southern part of India, whom we call as ancestral South Indian. Other one is ancestral North Indian. Probably there was second wave of migration from Africa towards Europe, and somewhere there's a split of the groups. One group has gone to Europe, other group has come to northern part of India, whom we call as ancestral North Indians. And there are several studies which I am not showing here have shown there are some mutations which are present in the Europe, Middle East, and northern part of Africa, particularly the skin color specific mutation, SLC34 A5G, and one of the specific haplogroup in Y chromosome, and also the lactate persistent gene mutation, which are common between Europe, Middle East, and other parts of Africa, which connects that hypothesis. And these two groups are mixed and gave rise to many population groups in India. On the other side, if you look at the principal component analysis, these are all different Indian populations. These three are map sample from Europe, Africa, and China. There are several hundreds of individuals all in this cluster, suggesting that genetically they are more homogeneous, right? Therefore, no such difference as you see here exists in all three map samples. Let us look at Indians. Every single population is unique. And although every population is unique, but still we are all connected genetically. So that shows that India is not inhabited by single large population. Rather, we can say India is inhabited by large number of small populations. Although every single population is unique, but still there is a connection between the population because of the admixture which took place. So one example, you take the skin color alone. India is the only place where you see extreme light color, extreme dark color. In between, there are several other colors, all kind of shades you can see. So that itself one example that there was an admixture took place, which I'll come a bit late to show that when this admixture took place. So these two groups are admixed and gave rise to many population groups as we see here. Subsequently, we want to address when this admixture took place. So we took large number of samples from south, large number of samples from north, and try to understand based on every generation during the meiosis and the recombination, right? Between the homologous chromosomes. Based on that, we try to understand when this admixture took place. And we show that both ancestral South Indian and the ancestral North Indians have initially gave rise to many population independently. You can imagine the initial stage when the first out of Africa human settled in somewhere in jungle after finishing some of the food material available in that place. Group of people must have migrated to other place in search of food, right? That's the way it happened. But after migration, there's no connection between these two groups. That this has been left alone, this alone left alone, then different groups have emerged. When different groups emerge, what happens that there's a marriage happens only within the groups, called as endogamy. Right? So that way, in the last 4,000 years, every single group has been independent. But last 2,000 to 4,000 years, these two groups are admixed. The color indicate how the genome is admixed. After admixture, every single population started maintaining the endogamy, means marriage. So this is much before that the caste system came into existence. So this gives you overall picture what happened in the past. There was a two founding groups. They gave rise to many population. They are mixed. Gave rise to independently many population who have been maintaining the endogamy for the last 2000 years. One reason could be because of the caste system scale around 1000 years back must have put more pressure on every single population to maintain the endogamy. How quickly I will take you through. So, what is the impact of this endogamy on our health and disease? So, again, we took another study. This time, we don't want to restrict only within India. We extended the study to other South Asian countries, including Pakistan, Nepal, Bangladesh, and Sri Lanka, and other South Asian countries. The reason is one of the studies where we have identified a mutation which is responsible for causing cardiac arrest in the myosin binding protein C2 gene. When we look for this prevalence, it's existing only in South Asian countries, but not in other countries. Due to time limit, cannot show that here. So I think whatever we do here, it's also relevant to other South Asian countries. Therefore, we do that. And you can see these are the places where the samples are being collected. When we try to look at it, to our surprise, came to know that this is not a population specific recessive disease are existing in India, which is not known so far. And we thought that it's a good opportunity for the next generation to look for population specific recessive mutation, which is responsible for causing population specific recessive diseases. So to give you very briefly, that this is called IBD, identity by descent score. Each line represents one population. We have used 275 population from South Asian countries and 2,800 individuals from all over South Asia. If the line is more, there are 275 lines. The line is more, that means the relationship between different individuals in the same population is much higher. Okay. So they share a common haplotype. That means those who are here, they have more chance of having population specific recessive mutation, which might cause population specific recessive disease. The reason why I'm saying, yeah, there are two populations which are being included here. One is Asian and Jewish, other one is Finnish population. These are small population, well studied, and there are large number of diseases are being mapped. You can see these are the name of the diseases, and you can see there are most of them are autosomal recessive, and these are the genes. Same way in Finnish population. If somebody has autosomal dominant, then whoever has the disease in heterozygous condition, we have 46 chromosomes, 23 pair, 23 male from mother, 23 from the father. If one of the pair has the mutation, it shows the disease as the dominant. That means every single individual who carries the mutation have the disease. That is obvious. But the problem here is recessive mutation. But the recessive mutation exists in a heterozygous condition. It cannot cause any disease. I'll explain you in the coming slide. So when you have such a large number of individuals having, we don't have any clue when we collected the sample until the section. So we went back to the population. Before that, I'll explain this. Pedigree, probably all of you are aware. Circle indicates female, square male, and you put the line that means there's a marriage between them. These two are their origin. The mutation, it shows in one of the pair that makes heterozygous. Therefore, there's no disease there. A husband doesn't have that mutation. Right? Then this particular mutation goes generation after the generation in a heterozygous condition. Until here, nobody have any disease. Right? But you can see the other side has come here. These two individuals are absolutely fine. But when they marry, there's a 25% chance that they contribute this abnormal chromosome to next generation, therefore the disease. So that condition is very, very high among Indian population. So we went back to some other population to see whether such diseases are existing. So particularly we identified a few population. And when we went back, and we found that one of the populations in extreme south near Madurai has a disease called junctional gyrate epidermolysis bullosa. So this is disease which causes skin rash and also congenital heart disease and after the internal organ. And these children die within 2 months after the birth. So you can see the pedigree. Parents are absolutely fine. We look for a known mutation, there's no problem. Therefore, we went on to do exome sequencing and identified a 11 base pair deletion that we call LAMP3. Okay, parents are heterozygous and the children are not surviving. But in this kind of scenario, but somebody goes to the population and look for any disease, there's no individual in the community existing with the disease. Because children with the homozygous born very rarely, and there's no one surviving, therefore you don't see. So this is one such case. Similarly, there are large number of cases are existing in India. And we initially compared that with the 10,000 genome sequencing, which Prime Minister online came and announced and released it. So this is a earlier one. I'm glad that the topic is there because one of the DPT officer who was involved in this particular study. And there was no individual from the 10,000 genome from 83 populations shown that mutation. When we look for the mutation, the global database, you can see 8,62,000 chromosomes as a 2,000 individual. And if you look for the chromosome level, more than 17 lakh chromosomes all over the world doesn't have, except 2 South Asian individuals in a heterozygous condition. This is such an ultra rare mutation. And if the mutation is very rare, what we do, we create model to look for whether this mutation is a real responsible for the mutation, responsible for the disease or not. What happened? This is a complete set of amino acids. This code for 1702 amino acids. This is where the mutation. The mutation is truncate the protein, become nearly 800, removes approximately 380 amino acids. Therefore, it's very serious. So we created the animal model using CRISPR-Cas, precisely deleting this 11 base pair. And these pups are not surviving beyond 2 days. The human survived 2 years, here 2 months. Therefore, we could not study further. However, within the 2 days, we can see there's a blister started appearing. And within 2 days, they died. And to our interesting thing which I want to highlight, sometimes when you do CRISPR-Cas, there's a non-specific, and interestingly, we found there was one multiple base pair deletion clone. Okay? And we continue with that. This is a 45 base pair deletion, this is an 11 base pair, but this is a non-frame sheet. Although the deletion is 45 base pair, much bigger than the 11 base pair, but it removes only 15 amino acids. Therefore, the severity of the disease is much lesser. We created the animal model. Continue watching it. Till 4 months, there was no symptoms. But after 4 months, slowly started the symptoms. I think 4.5 months. And after 6 months, you can see this is a homozygous 45 days deletion mice. You can compare how small it is, and the internal organs, and you can see the heart is enlarged, dilated, spleen is enlarged, stomach becomes very small. We also did extensive study. Some of the things which I showed, many of them I did not keep it here because of the time. But look at the skin, there's an impact dermis and epidermis. Whereas if they cultivate the epidermis, you can see there's a gap between dermis and epidermis. This is where the blisters start appearing. We also looked at the micro CT of the level-based fermentation mice, very tiny thing. This is the wild type, is a heterozygous, both look same, but look at the level-based homozygous, you can see hypertrophy, 8%. There's also a hole causing the septal defect, is a cross section. And we also created homozygous line for the mother's blood because homozygous children are not surviving. And I'm not showing that. I will show you this particular slide that when we do something in the lab, how we can extend that to population level and the society level. So in this case, the mother was one of the mother was pregnant who lost a couple of children due to level-based fertilization. Brought the mother to Chennai, collected the antibody and tested whether the fetus carrying wild type or homozygous mutant or heterozygous. And we found that there are two alleles, it's like heterozygous, and we also confirmed by Sanger sequencing. And allow that fetus, advise them to allow the fetus to grow, and it's child born about 1.5 years back, with, surviving without any phenotype. So this is one aspect. Now subsequently we approached the government, government to give us the permission to screen the entire population. There are about 5,000 individuals. Therefore we can give prenatal counseling, premarital counseling, and if you do that for several years, we can actually eliminate this location and there won't be any such children in the future. Just to justify that, I'm talking in the how many minutes? Yeah. So, yeah, I'm going to justify that I'm speaking in the fishery conference. So this is one of the mitochondrial disorder, mitochondrial neurogastrointestinal encephalopathy. They're all multiple symptoms. Again, it's a novel mutation in TIMP-G. You can see wild type, homozygous mutant, and the heterozygous. We have both G and A. So we created zebrafish model for this by silencing using mapillino. You can clearly see there's a muscle loss, and you can clearly see that muscle loss, edema, and also the autism loss, mimicking the phenotype which we have seen in the patient. So having seen all of these, and I was in CD early getting that. So we had a mission program again, it was part of it from the side. And there are 16 laboratories who are actually doing their work, has been part of it, and there are a lot of activities, including sequencing, and, counseling, predicting whether the mutation, novel mutation has any role. We have 4 different model systems, and also we want to have therapy, particularly for neurological disorders. And we created 8 videos in 8 different languages for making awareness. If somebody is interested, you can go through this. And if you come across any such disease, you can also inform CDFD for extending the study funded by ABP. So with that, I thank all of those who have involved in this study, like Professor Lalvising and all my colleagues. Without extensive collaboration, this work is not possible. Although I told very briefly, but there's a lot of work involved there. And also, India, Brazil, and other funding agencies. Thank you very much.
Okay, it was an amazing lecture with the very broad overview of the genomic evolution of Indian population to the current ongoing project of the 10,000 genome project. It's so many fascinating outputs. I think we're really very short of time now to take more questions, but please do talk to Dr. Thangaraj in the next time. But conversation for taking lead into some of the very technical qualified. Thank you very much.
Stop share.
All right, so our next keynote speaker is Professor Gopal. He is now studying, career, PhD in molecular biophysics from IIT Bangalore, MSc Physics IIT Kanpur, and a postdoc from NIMR London, and then a professor of structural and biology since 2023 in the professor here. And then, of course, was earlier, different positions. They got 6 patents, more than 100 publications, something so but never awarded. I mean, all those speakers, they are so outstanding. And Anand Birla Science Prize and of course the DBT National Bioscience Award. And he's a fellow of all the academies, INSA, NASI, DC Ghosh Fellow. And this is a very DC Ghosh Fellow, very prestigious, it's a very prestigious fellowship. In fact, it's not a fellowship, it's a big title, so maybe it's fellowship. And then, of course, he is the chair of the drug discovery platform. And then, he's also secretary of the Indian Academy of Sciences. I think this is the state. So we have the secretary fraternity now. So I'm the secretary of the National Academy of Agricultural Sciences. Okay, should be more closer now. Member of the governing body, DBT, rather than the Center for Biotechnology and many others. I think so. We invite Professor Gopal for your talk. Thank you.
So thank you, Professor, for that introduction. It's actually coming from him. It means a lot. Anyway, thank you for all the organizers, especially Goswami and others. And so in the next 15 minutes, because you all are hearing me for a short while, what I thought is I'll give a feel as to where an area is coming up and why I think it matters a lot, even for us who are making the transition, and for institutes like these, where you're talking about fisheries and fishery studies and taking it forward. So I've put this title this way: Design of Synthetic Biology Tools for Engineering Biological Systems. So, if you wanted to start off, the first is more of a question. With the molecular and structural biology in the year 2025, that's where we are. So when you look at it, structural biology by itself, looking at structures of proteins, DNA, larger complexes, this had a fundamental change in the year 2022. Google, the shadow company called DeepMind, essentially used AI and the tools where you can generate models for almost all the structures possible in the living universe. Then you may ask, what is it that's new? What's new is that we may know the structures, but actually, after that information, you probably begin to have a whole new set of questions for which you don't have answers. So, what is it that I think are the open areas and where we are on it? So, for today's part, because I have only about 10 or 15 minutes, I'll focus on a very narrow component and then probably hint at the directions that you might see how you could stretch it forward. Because of Lakrasab's introductory talk in the morning, I put this title. He asked me to define biotech. I would say harnesses life's existing mechanisms. Biomimetics, it mimics nature's design. Synthetic biology, on the other hand, can engineer new biological systems or modifying existing ones. So this is something which is not exactly something that we guys have figured out in 2025. If you were to look at it, way around in 1680, people could talk about or ideate about these things. Why would fishes stay underwater, they said? Because you have these compartments where you can adjust the air pressure, and therefore you can now go from depths about 500 feet to 1,500 feet, go slightly below, you get punctured, go slightly above, you have other partial pressure issues. So how does one treat this? So why develop synthetic biology tools? As you can see out here in this background, the sugarcane. You may say, what is it that sugarcane has for this audience? So I've taken this particular thing, which is it's got a generic title. It says the hideous price of beauty. What it says is you have this compound called squalene. And if you don't believe in it, even as you enter the Adani airport outside and you see this poster board, and it says about the skin peel that comes in. You have orange peel on one side, and you have the skin ointment on the other. That's squalene. You know where it comes from? It comes from sharks. Now, so you have so many sharks which are getting killed. And so, for example, this data that's presented from this site, I could not validate it. I've taken it straight from the site, which says that this is the kind of numbers that you need to feed. So what does synthetic biology do? When you have such compounds that are greatly in need, you identify the entire pathway, stitch it into the molecule, into your chassis of interest. So I'm bringing in some terms that are new to synthetic biology. We don't refer to genes as genes, we call them coding components. We refer to enzymes as parts. We refer to the systems as chassis. So in my lab, for example, we work on bacterial chassis and yeast chassis. And so in some sense, you can start modulating the chassis based on what you want to do. So is it all that straightforward? So once, and this is where I thought I should give you a feed as to why biology is still very interesting. When I say synthetic biology, you may get a feel that this is a man who's almost, he comes from Bangalore anyway, so most likely he's designing a two-wheeler with an electric motor. Not really. Out here, the biological system evolves. So if you try to make some changes, you compensate on another part, and that's called self-fitness. And that's something that the first speaker also alluded to. So what we've now begun putting our thoughts into is design simulations of entire cells. We work on bacteria and yeast cells for a start, but presumably as compute power and our mechanisms grow, they become better and better. So what you now have as this variable, environmental conditions, altered expression levels, selection pressure, self-tolerance. For you and me sitting in this audience in the Fish Institute, you may wonder what is it that means. Remember, as you go lower and lower in depth, your environmental conditions become quite interesting. So what you now have is cyclophiles, mesophiles, thermophiles, you have variations in the enzyme. Then you have variations which are acidophiles, which means you now have entire enzyme systems that can work under very different conditions. What we then do is try and build up these models. This, in some sense, is a black box. Remember, even your chat GPT, even all the other tools that you use, work on the same black boxes. And one of the things that is sadly missing right now is good annotated data. So if one were to think about developing programs in this direction, I suspect getting clean data for the machine learning is something that's absolutely essential, which is why people like us still work with simple unicellular systems. Even there, the variability is quite intense. What we then do is try and see if you can analyze a flux of a given metabolite. So think about it. A chassis is a set of molecules. It has the genes, it has the enzymes, it has the different components and the nutrients coming in. You have metabolite fluxes which will go from one to the other. So essentially what you now have to have is a way in which you can put all of this into differential equations and put nonlinearity. And then ask, how does this vary as a function of time? And this is something that can then be experimentally tested out. One, for example, a very simple example, but you realize how awkward it is. E. coli. Now, almost if you walk with me to any biotech sector, and you would have E. coli and yeast as a model systems. And you would also be surprised to figure out that 9 times out of 10, they have fundamental issues of batch-to-batch variations. You know why? It's called evolution. As you put pressure, the E. coli now suddenly begins to behave very differently. So here, for example, we've now begun to look at E. coli strains that can be modified, where you get this natural things put in. So one of the ways in which people, and in fact large companies, evolve among them and so on and so forth, have invested probably millions of dollars trying to figuring out how to get directed evolution to work in silico. Now, for example, would we be able to leapfrog where we are? So one of the things that I still think for the younger people in the audience, and I hope that you might take the idea, is AI ML tools can probably substitute a lot faster, but In the absence of experimental data, that's still widely missing. So here, for example, you simulate classical enzyme fluxes, and then you ask, what is it that would work and what is it that may not? To summarize this particular slide, the take-home message is simple, straightforward knock-ins and knockouts are a bad idea. That's something that we figured out. And so in some sense, if you have a combination of more than 3 or 4 knock-ins or knockouts, you are naturally going to lead your cell, and presumably by extension your organism, into a fitness issue. Because you now have selective pressures that we'll have to accommodate for. And this is something that we've been also trying to feed back into our iterative cycle to learn. Remember, we are still learning, which means you make an error, you figure out and you simulate the error, and you learn again. And so, of course, simulations followed by actual experiments, and which of course would mean PhD students typically take about 5 to 6 years just getting the simulation data outside. What are the tools that I talk about? A very simplistic idea of a tool. You can start, what is it that we are worried about? You make too many knock-ins and knockouts, you have cellular fitness issues. What happens if I can just alter the expression levels to see how things might be? How did we get into this idea? It's different. So I started my career working on pathogenic bacteria, not on any of these. So if you take a mycobacterial cell inside the lung, and you ask, what's the expression profile like? 15 years later, you ask the same question, except that you have RNA-seq data, which means that you can now start learning from this and say, oh yeah, this is a cell, a mycobacterial cell that's encountered rifampin at this particular concentration to give you these changes. And I've learned, and you iterate. That's exactly the model that we are trying to put in for synthetic biology. So here, for example, is one set of tools that we've developed. Take a simple transcription initiation factor, which looks something like this. It's called this long linker. Now, what this linker does is it also now goes and works with the DNA. So you can now stay with the promoter DNA. You try to test it out on an E. coli system. Yeast still doable, E. coli kind of doable. With larger organisms, we haven't really been able to get very clear on it. So what we do is we start replacing some of these loops to create a set of synthetic parts. One example of a synthetic part would be something like this. They keep the chassis the same and just replace one with these set of linkers. So what you are doing now is altering the way in that same transcription factor would sit on one gene, you're now going to make it sit on something else. Or alternatively, make it sit on the same gene but change the expression level. Both of which will achieve the same goal. Now think about it in a combinatorial fashion. I changed this linker and I changed this loop. And if I have a variable of 10, I see 10C2 combination of spare parts that I can now use to engineer a cell. So what does this all lead to? What it leads to is some kind of correlation maps that we are talking about out here. This is a lot of data, but all that I would like you to appreciate is you can now correlate a sequence information with a DNA recognition. Experimentally validated using one form or the other. We use a variety of assays to do it, and some of them are intra and some of them are in vitro like this, and you can get some readouts which give you some clean parts like this. Suffice to say, you now have a set of transcription initiation factors, probably 100, probably 1,000, and you now ask the question, can I use it to now start doing something new? So this is another variable that we've introduced out here. What we would now do is examine what is the variable if I were to change this linker. Why did we even go for natural linkers? I should tell you that. Remember, nature has evolved each part for a function. So if I were to now replace this with a random polypeptide, it's flat. Nothing works. Which means I can choose the nature's spare parts for stitching into that particular sequence. And if I were to choose this from a cyclopile, I get a different sequence. You choose it from a different other organism, I get a different variation. And so in some sense, as you can see, I can now get a variation in input also in terms of expression levels. So what did I achieve? You had a transcription initiation factor. I could change with DNA it would make. I could also change how much of the DNA it would make. So I can modulate the entire system purely by looking at these components. What did we manage to get across? We don't change the components of the genome at all. we only change one base or one group. How does this work? I told you about squalling. I give you a very simplistic example of making these. These, for example, are phytochemicals. You could also use this for making other kinds of chemicals. And in fact, for those of you all who would be interested in synthetic biology applications, you may be surprised that this is pretty much what the BioE3 policy is all about. Trying to make more of these so that we cut down on the natural sources, maybe plants, maybe animals, and definitely marine life. So if you wanted to make one such, a very simplistic example, you start with a two enzyme systems. You now know the variation in this two enzyme system. This is how we start. Characterize the enzymes, and then you figure out what the rate limiting step is. Then you start the second step in the pathway, where you can introduce these enzymes either within the genome or external. Then what you do is you ask a question, how much? So the rate limiting enzyme, you make it in much higher quantity. And what you do in that sense is you reduce the stress on that particular cell that is making this particular product. Is it successful, you may ask? You talk so much. Actually, you'll be surprised. It actually does. Which means you haven't changed any of the genomic components, by just changing the transcription on/off switches, you're able to get apparent increases in this. And these also now work quite well when you want to scale up from 10 liters to 1000 liters to 1 kl to 30 kl. And that's the kind of fermentation products that go on. So what does synthetic biology mean after all of this? What it means is you can study all of these in very different ways. Another synthetic biology tool that we are working on, as a very quick example, and this is something which I hope some of you all who might be interested. Fishes, especially those which come under very deep levels, are essentially cyclophiles. You see, the enzymes that are there in cyclophiles have very different dynamics. Enzymes, by definition, are not the lowest energy state. They're supposed to be quasi-stable, which means they can have dynamics and they work. And for an industrial application, having an enzyme which comes from naturally optimized at that layer is actually very useful. So what we do these days is we look at those sequences, and then we also try and start making variations to look at the dynamics of the scheme. So in this particular case, what we are talking about is taking a cyclophile is low temperature, which means that you can do the same reaction which you otherwise had a problem with compound stability to come in at an optimum temperature. So think about it, the possibilities are immense. So what did I give you? I hope I'm on time, Sanjeev. So to summarize, what have I said in the last 15, 20 minutes or so? I've given you a field of synthetic biology where you can use transcription factors as on-off switches. What I've told you about is that the possibilities for these using natural sources are immense, which means if you had a fantastic collection of natural compounds, you could just sequence it and start playing with it. Imagine if you had the sample coming from the northeast, if you were looking at terrestrial samples. The genomic sequence variations is phenomenal, most of which are not known. So you have a natural set of parts which you can play with. So what have we learned so far? We've taken this area up in the lab in the last 7 years. So typically, I'm now about 25 years of service. Typically, every 10 years, I've switched fields because that's important to learn. What we've now done is we've moved more and more into computing because AI/ML is, we think, where biology would be moving. It's a fact of life. And in some sense, molecular biology, 10 years from now, most likely will be more computing and less experiments because it will be guided. Second is we now are getting tools which allow you to figure out what are the effects of knocking out and knocking in, which means the good old-fashioned thought, oh, this is an essential gene, I should not touch it. This is not essential, let me play with it. That's gone. So in some sense, what we also realize is some genes which are non-essential are also there for a certain self-witness function, which is difficult to analyze. And the third is about simulations again. And this is where I've talked about designer enzymes. And at some point of time, if you're interested, I could also show you some work that we've done on some of these enzymes. All of this would not have been possible without generous funding. So the work that I do essentially comes from the DST and the DPT. This is something that supported me over the years in the lab, of course, 24 years. There are co-workers who spent a lot of time generating some of this data. I've only named a few of them because I've selectively removed all the other model systems to summarize what I've told you. All the computing, I've been doing it at IIT Madras simply because Karthik and his group are much better at it than we ever are. And honestly, I could not have done anything had it not been for the Indian Institute of Science. That's about it. Thank you.
Thank you, Dr. Gopal, for giving a entirely new concept of synthetic biology now. And the new dimensions of synthetic biology, the new models, new wonders of synthetic biology and bioengineering, including how you can create the factor, how you can alter the messenger RNA proteins, and how the simulation possibilities, for cell engineering. It's a wonderful. Thank you so much. And thank you very much. What is the announcement?
Okay, thank you so much, sir, for sharing this session. And Dr. Sandeep Srivastava. And a very important session. Three keynote speakers, one from our university related to genomics, genetics, and biotechnology of this. And then these two are again very quite interesting in general. And then it's quite interesting so that we can do some collaborative works, and therefore I am looking forward to that collaboration. So now I request Dr. Sir to honor our keynote speaker and co-chair. Sir, please.
Sir, please, sir. Okay. 1 minute, sir. Thank you, sir. Great, sir. Thank you, sir. And I request Professor to honor Professor. Thank you. Yes, sir. Sir.
Now I request sir to honor our co-chair, Dr. Sandeep Srivastava.
Sir.
Okay, now I request our director sir and then deputy director general medical science, respected Dr. Raghavendra sir, please honor our chair.
Sir, you can have a. Please, sir. Sir, we have a one. Yes, sir. Thank you.
Now, thank you very much. Sir, now we break for lunch. Then after lunch, the technical session will be there in respective hall. Maybe 319, 428, 418, as per the technical problem. Thank you very much. Presentations are after lunch, immediately after lunch. Respective rooms, please note your presentations. Yeah, maybe we have to be
Please, sir, you can have. Sir, please.
So we'll be here by 2 o'clock after lunch. And
I know.
Around 2 o'clock, we will be the respective one for next. Thank you very much.
दोनों ग्रुप ये और वो उसमें दोनों को ये करके रखिए तो अच्छा रहेगा। Online यहाँ कुछ नहीं है, ग्रुप फोटो का वो लगा दो ना अभी जो खींचा था, लेकिन यहाँ कुछ है नहीं ना, नहीं होगा ना, कुछ ना कुछ चलेगा। हाँ जहाँ है उसको लगा दो ना। यहाँ पे भी है पूरा assessment now வேணும்னு செய்யறது I'm based in Delhi. Right, இங்க வர அப்புறமா பேசுறேன். இல்ல நீங்க பேசுங்க. You want us to stand here? Sure. We'll plant it. Yeah, thank you so much. Thank you. You do very well. You do a very interesting. Just put my name, Gopal. I saw your. I would not take the. I would cameraman was standing.
Actually, I things.